Right, there we go. Yeah, perfect. Yeah.
Sure. Morning, everyone. Welcome to day 2 of our World Antimicrobial Awareness Week Symposium. We are really glad to have you back. We hope you enjoyed day 1 and learned a lot from day 1. We had really good keynote speakers in day 1 in different areas to do with antimicrobial resistance. We also had some really fantastic presentations as well from our researchers and students. And I think that is also what's going to happen for day 2. Just apologies also for starting late. I had some connection issues, and so that is really my fault, not the fault of the organizing committee as well. And then just to let anyone know who's joining us day 2, I'm Dr. Yunusa Jaswa. I'm with Water Research Commission, where I'm a research manager there. And together with Dr. Klope and Dr. Nonhlanhla Kalibaila, we run the antimicrobial resistance and emerging contaminant program in the WRC. So this area of research really spans the whole of the WRC, and we have been involved in this symposium. I think this is our 3rd or 2nd year. I think it's actually our 3rd year running now, and it's been fruitful every time we've run this process and this symposium. So without taking much time, I want to welcome you all once again, and also just to introduce our keynote speaker, the first one for this morning. I am just going to go through his biography quickly, a short bio. Dr. Brandon Reinecker obtained his PhD in microbiology from Stellenbosch University in 2020, and he's currently a postdoc in the Water Resource Lab that is being headed by Prof. Khan at the same institution, which is Stellenbosch University. His research focuses on water treatment systems, molecular viability assays, QMRA, quantitative microbial risk assessment, and then antimicrobial resistance, as well as the use of bacteriophages as biocontrol agents. To date, he has published about 19 peer-reviewed journal articles, a book chapter, and he's also the first author of 8 publications. He also currently supervises a couple of honors students, 1 MSc, and then is also supervising a PhD student as well under Prof. Khan's research group. So with that, Brandon, we're looking forward to hearing your keynote this morning, and when you're ready, you can please go ahead. Thank you.
Thank you very much. I'm just sharing my screen.
Sure.
Is everybody seeing my screen?
Yes, we can see it.
Okay, thank you very much. Good morning, everyone. So today I will be presenting a brief overview of the research that is being conducted in the Water Resource Laboratory that is headed by Prof. Wissal Khan. And basically, we just want to give you guys a brief overview of how we are trying to combat the threat posed by antimicrobial resistance using surveillance, risk assessment, and biocontrol. So following the presentations of yesterday, and hopefully by the end of the symposium, we should all be aware that the significant increase in multi-drug resistance amongst bacterial pathogens poses a serious public health threat, as treatment options to combat associated infections have become limited. Now, although numerous bacterial species have developed reduced susceptibility to antibiotics, a particular group of pathogens, commonly referred to as the escape pathogens, namely Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Cinetobacter baumannii, Pseudomonas aeruginosa, and lastly, Enterobacter species, contribute significantly to the burden of disease in both developed and developing countries. And this is primarily due to the increased propensity to carry multiple antibiotic resistance genes. Now, worryingly, the emergence of antibiotic resistance is no longer only a problem in clinical settings, as the misuse and overuse of antibiotics in various sectors of society have contributed to the dissemination of antibiotic resistance bacteria into the environment, and more specifically into environmental water sources. So thus, while the majority of multi-drug resistant escape pathogen outbreaks have been linked to hospital-acquired infections, community-acquired outbreaks have also been linked to the escape pathogens, with environmental water sources highlighted as a potential escape pathogen reservoir. Now, this then brings us to one of the main projects in the Water Resource Laboratory, which aims to investigate the prevalence and antimicrobial resistance profiles of escape pathogens in environmental water sources in urban and rural informal settlements of South Africa. And this primarily forms part of the research being conducted by Ms. Julia Dennison. Now, this research is important as many of these contaminated water sources are used for sustainable agriculture and community gardens. Moreover, waste removal and sanitation infrastructure is usually lacking in these non-sewered informal settlement communities, and the risk of exposure of community members to the escape pathogens is thus increased and must be characterized. So the second part of this project, we are then aiming to assess the health risks posed by these environmental water sources to community members based on the presence of the escape pathogens using quantitative microbial risk assessment. Now, this is a WRC-funded project where Prof. Wissal Khan is the project leader based at Stellenbosch University, and we are also collaborating with the Water Health Research Center at the University of Johannesburg. And because we're working at two institutions, we then also have different sampling sites. So for Stellenbosch, we are collecting samples from Flottenburg Informal Settlement and Salarispaas Village, while the UJ research team is collecting samples from Zandspruit Informal Settlement and Tembisa Informal Settlement. Now, there's also a brief deviation in the methodology that we are applying because we're based at two different institutions and have access to different equipment. But we are performing validation steps during the study where samples are being sent between the institutions for confirmation. But overall, we are isolating the escape strains from various environmental water sources, and this includes stream water, river water, surface runoff. At the University of Johannesburg, they've also collected some tap water, grey water, and stored water. And we are then culturing the escape strains using selective media, and this is then followed by isolate identification and strain differentiation. We are then moving over to assessing the antimicrobial susceptibility of the isolates, and then we are also testing the colistin susceptibility of the gram-negative, multi-drug resistant and extensively drug-resistant isolates. And then lastly, we are going to try and elucidate the resistance mechanisms that are being employed by the MDR and XDR pathogens by screening for various resistance genes. So what have we found? Now, I will only be highlighting some of the preliminary results for the SUN research team, but in total, we started off with 537 presumptive escape isolates following the culture-based analysis using selective media, with the presumptive Enterococcus faecium and Klebsiella pneumonia accounting for 47% of the isolates. Now, when looking at the distribution of the samples or the isolates per sample type, overall lower isolation frequencies were obtained for the surface runoff samples, while the primary water bodies such as the marsh and stream water sites exhibited higher isolation frequencies. Now, it's important to note that although the surface runoff samples are more regularly contaminated by pollution and anthropogenic activities within these communities, the grey water that is commonly discarded down this waste stream regularly contains cleaning agents and chemicals, which may influence the survival of the escape pathogens. We then moved over to conventional species-specific PCR assays to confirm the identity of our isolates from the different sites. So at this point, I would just like to note that we are in the process of optimizing the Intravector species PCR, and then also the PCR for the Staphylococcus aureus. However, we did run a Staphylococcus genus-specific PCR to try and narrow down our number of isolates. Now, based on the PCR analysis of the presumptive escape isolates, 18.2% to 68.5% of the isolates were PCR positive for the correct species. It is thus important to further characterize and this highlights why it's important to further characterize any presumptive isolates using molecular methods such as PCR and sequencing, as your non-target bacteria may also be isolated along with the target bacteria using selective media. Nevertheless, these results illustrate that the escape pathogens may have environmental reservoirs outside clinical settings, which enables them to survive and proliferate in the natural environment and may subsequently aid in spreading these organisms within communities. Now, in order to determine the similarity between the environmental escape isolates, molecular genotyping was conducted using RET-PCR analysis, and dendrograms subsequently generated using bionumeric software. And basically, this enables us to look at the DNA fingerprints of the organisms to try and see how similar they are to one another. So over here, you can see what the analysis looks like. So this is the preliminary results for the Klebsiella pneumoniae isolates, and we had 7 distinct rep PCR type strain groups, and then also 7 singletons, which were unique and did not fall within a specific group. So the singletons are indicated here in the bottom right-hand corner. Now, following the rep PCR strain differentiation, we were then able to select representative isolates from each group and some singletons that would also then be representative of our different sampling sites and sample types, and use these isolates for the antimicrobial susceptibility testing. So we went ahead with the Enterococcus faecium, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. Now, as I mentioned earlier, the escape pathogens are notoriously known for their role in life-threatening nosocomial infections and the high levels of antibiotic resistance, which stems from their increased propensity to acquire antibiotic resistance. Thus, while the culture-based analysis and the PCR-based assays enable the elucidation of the potential environmental reservoirs that may harbor these escape pathogens, it is essential that the antibiograms of the escape isolates be determined. We then started off with the Enterococcus faecium, and based on the disdiffusion assays, 4 Enterococcus faecium isolates were classified as multi-drug resistant, and 1 isolate was classified as extensively drug resistant, displaying resistance against approximately 67% of the antibiotics tested. For the 10 Klebsiella pneumoniae isolates we have worked with thus far, 3 were identified as multi-drug resistant, while no XDR isolates were identified. In comparison, 1 MDR, 1 XDR isolate was identified for Acinetobacter baumannii, with the XDR isolate displaying resistance against 78% of the tested antibiotics. And lastly, no MDR and XDR isolates were identified for the Pseudomonas aeruginosa isolates analyzed thus far, but we are still busy with this. So following the antibiotic susceptibility assays, the MIC and MBC for colistin were determined for the gram-negative MDR and XDR environmental isolates. So the MIC for both of the Acinetobacter baumannii isolates were 2 micrograms per milliliter, while the MICs for all 3 of the Klebsiella pneumoniae isolates were 8 milligrams per milliliter. The K pneumonia isolates were then classified as colistin resistant based on the CLSI and UCAST 2021 MIC breakpoint guidelines, while the Acinetobacter baumannii isolates were intermediately susceptible to colistin based on the CLSI guidelines and susceptible based on the UCAST guidelines. So to conclude this part of the aim of the project, we are in the process of screening the MDR and XDR isolates for various antibiotic resistance genes to try and elucidate the antibiotic resistance mechanisms that are employed by these escape pathogens. So the detection of the MDR and XDR escape pathogens in water sources that are used by community members, such as the stream water sample we have here in the center, which is commonly used by community members for washing laundry by hand, is a significant concern as the dissemination of the antibiotic resistant escape pathogens in these environments may increase exposures of communities to these contaminants. It is thus crucial that the potential health risk these environmental water sources pose to community members be quantified, which brings us to the second part of the project, where we are going to apply a quantitative microbial risk assessment approach. Now, in order to perform QMRA, 4 stages of analysis need to be conducted. This is starting off with the hazard identification, which in our case is the escape pathogens that we will be quantifying using viability qPCR, and then dose response modeling, where we take into consideration how easily the target organism causes infection. We then move over to the exposure assessment, where we take into consideration how the community members come into contact with the water sources and whether the exposure leads to ingestion, inhalation, or dermal contact with the water. Lastly, we perform the risk characterization, where the combination of all the information allows us to generate a prediction-based analysis model, which elucidates the potential health risks that correlate to specific pathogens based on the exposure scenarios that we tested. Now, we have applied this methodology previously to quantify the risk associated with using rainwater for various domestic activities. And this is just an overview of what the results would look like. So on our y-axis, we have the risk of infection. So the higher up on the y-axis you go, the greater the risk. And the red dashed line indicates the benchmark limit of 1 in 10,000 individuals getting infected while performing that activity. So for example, in our previous research, we showed that drinking rainwater exceeded the guideline limit, while using the rainwater for washing laundry by hand was within the guideline limit, and thus presented a reduced risk of individuals getting infected. Now, while we are still in the process of quantifying the health risk posed by the antibiotic-resistant bacteria, the one thing we do know is that the widespread dissemination of antibiotic-resistant bacteria in the environment that may lead to community-acquired infections represents a new challenge with important implications for the effective treatment of infection. Now, any treatment strategy that you would then want to apply to target antibiotic resistance would thus need to adhere to a One Health approach and be implementable for animal, environmental, and human health. So in the research group of Prof. Khan, what we are trying to do is use biocontrol, and more specifically using bacteriophages, predatory bacteria, or biosurfactants to target antibiotic-resistant bacteria. Now, as my time is running out, I would just like to highlight some key points with each intervention, starting with the use of biosurfactants. So biosurfactants are amphiphilic compounds that are non-ribosomally synthesized by various microorganisms, and they can be divided into groups such as glycolipids and lipopeptides, with their antimicrobial mode of action then primarily linked to their ability to disrupt cell membranes through pore formation. Now, in our research group, we have looked at the biosurfactants produced by Bacillus and Pseudomonas, which formed part of Tando's PhD and postdoc, and then also Serratia species, which formed part of Tanya's PhD, and then Lara was also working on it this year with her honors. So our results then indicated that these compounds are effective against pathogenic and antibiotic-resistant bacteria, and also have biofilm formation and disruption capabilities. Specifically, the lipopeptides were effective against multi-drug resistant and extensively drug resistant Acinetobacter baumannii, Pseudomonas aeruginosa, and Klebsiella pneumonia, while the rhamnolipid was highly effective against Listeria monocytogenes, which is an important foodborne pathogen. Additionally, Tanya found that Serratia marcescens was producing various secondary metabolites, and she is currently busy characterizing these compounds using molecular networking and a metagenomics approach, and also assessing their antimicrobial activity. Now, our second biocontrol intervention is the use of predatory bacteria, and depending on the predation strategy they use, they are also sometimes referred to as vampire bacteria. And we have Monique working on this project as a postdoc, Ben as a PhD student, and Megan as a master student. Now, specifically, we are looking at Bordetella vibrio bacteriovorus, which is a highly motile, small gram-negative bacterium that is able to infect other gram-negative bacteria, and then subsequently use their nutrients to replicate within the host cell, and then they burst the host cell upon release of the progeny. Now, thus far, we have isolated Bordetella vibrios with activity against Klebsiella pneumonia, Acinetobacter baumannii, and Pseudomonas aeruginosa, while we have also looked at the survival strategies employed by the gram-positive escapes to invade infection. Now, the current ongoing research is looking at the interaction of the Bordetella vibrios in mixed microbial communities, and then also looking at potential resistance or persistence mechanisms employed by the prey organisms, and then also looking at how we can combine the biocontrol treatment with other physical treatment strategies. Last, but certainly not the least in our research group, is the application of lytic bacteriophages as biocontrol agents. This is a project where I'm working together with Tinta Morris. And now lytic bacteriophages are bacterial viruses that infect their host, replicate inside the host, and then subsequently lead to host cell lysis upon release of the progeny viruses. And this is just some electron microscopy images of what our bacteriophages that we have isolated look like. Now, all of our bacteriophages thus far belong to the Cordoviralis order that is divided into the Podoviridae, Myoviridae, and Syphoviridae families, and they are normally characterized and differentiated through their tail structures. Now, overall, we have isolated bacteriophages with activity against Klebsiella pneumonia, Pseudomonas aeruginosa, and Enterobacter cloacae. And then what we've also started to do is look at the biofilm disruption capabilities of the bacteriophages, and then also the potential synergy between bacteriophage antibiotic combination treatment strategies, with our preliminary results indicating the increased susceptibility of the antibiotic-resistant bacteria to certain antibiotics following bacteriophage treatment. Now, in conclusion, the surveillance of antibiotic-resistant bacteria is crucial to accurately assess the potential health risks posed by these pathogens to communities, as the risk is no longer only limited to clinical environments. Lastly, treatment interventions that are applied need to take the principle of One Health into consideration to combat antimicrobial resistance from multiple fronts. So yeah, that was just a brief overview of what we do in the Khan Lab to try and target antimicrobial resistance. Thank you for listening. I would just like to take this opportunity to thank all of our collaborators and then all of the funding organizations for funding Prof. Khan's research, and then also the organizing committee of the symposium for the opportunity to present an overview of our research. Thank you.
Thank you so much, Brandon, for that. Are there any questions for Brandon? Or any comments in the chat? I don't see anything. Brandon, I think just from me, I was really interested by the levels of Klebsiella pneumonia that you were picking up in the environment, especially in the surface stream. With the escape pathogens, Klebsiella is something that's really not focused on, or even if sampling is done, it's really limited to one environmental matrix. And I thought that that's a nice result to really motivate for it to be looked at from a One Health perspective, you know, in terms of different environmental matrices and farms, manure, and then relate that back to clinical cases that we are seeing. Because in South Africa, we do hear quite a bit about Klebsiella clinical cases, but we don't really relate them back to what we are seeing in the environment. So yeah, I think that's a nice result showing that.
Yes, thank you very much. Interestingly, when we started off with the research, Prof. Khan started off with rainwater research, and Pseudomonas aeruginosa and Klebsiella pneumonia were some of these opportunistic pathogens that we were detecting in the rainwater. And then you would hear about all of these clinical outbreaks, and I think that is why Prof. Khan then just decided we need to look at the environmental reservoirs, because unfortunately it is something that is lacking in research. It has increased in recent times, but it's definitely something that we need to take into consideration, because you have all of this widespread dissemination of antibiotic-resistant bacteria into our water sources. Community members are coming into contact with it. And then, unfortunately, these organisms also have the ability to proliferate within these environments and then establish themselves in these new reservoirs. So it is definitely something that we need to take into consideration. And that's why I'm actually looking forward to the quantitative microbial risk assessment that we need to get to. And this is work that we're conducting in collaboration with Arizona State University to see how we can quantify the risk associated with these organisms in the environment, but then also taking into consideration the level of antibiotic resistance that they display, because that's also something that is not necessarily taken into consideration. A lot of the dose response models that we are seeing are either lacking or from the 90s or 80s based on mouse models, and not necessarily taking into consideration how these organisms would cause infection in communities, where you then also have a high proportion of the community members that may be immunocompromised as well. So these are all of the different types of things that we need to take into consideration. And you also can't just focus on clinical environments when you have, from an agricultural sector or from an animal point of view, these pathogens also being released. So yeah, I think it's definitely interesting work that may contribute a lot.
Yeah, and it enables us to really focus on the right pathogens, because I think sometimes we just spread our net wide, maybe because of what general international literature is saying, but as our local context, we need to know what to focus on. So definitely the escape way of work that we're doing is we are on the right track with that. I just want to check.
And thank you for the WRC for funding the project.
You're welcome. There's a comment from Chika, and it says, Brandon, there is no existing water quality guidelines for ARBs. So how are you going to consider ARBs in your risk assessment?
So like I mentioned, this is the work that we are doing with Arizona State University. And what they're basically doing is they're trying to see how we can incorporate the component of antibiotic resistance into the risk assessment model. So this will be based on assumptions, indicating, for example, that if this organism is resistant to this antibiotic, or resistance to this antibiotic has perhaps shown that the organism is able to cause greater infection or something like that. So it's not something that where we have concrete guidelines that we can go to. Our risk assessment will be based on the work that is being conducted at ASU at the moment, where they're trying to see how they can incorporate the antibiotic resistance displayed by these escape pathogens, and how we can perhaps adjust the dose response model. So for example, a Klebsiella pneumonia that is not antibiotic resistant would cause, will have a value of, just for interest sake, of 0.1 that we incorporate into our model. Whereas the same isolate with antibiotic resistant, that value would increase. So your risk would increase. So there's not concrete guidelines that we can use at the moment, but it's all based on these assumptions that we're going to bring in based on the research that they are conducting now.
Yeah, thank you for that, Brandon. And just to add here, a lot of the emerging contaminants we're working with currently do not have water quality guidelines, but that's why this research is also important, to be able to guide the development of that. Okay, thank you, Brandon.
Thank you very much.
Let me see. Oh, okay, there's one more question. Maybe you can just respond to that quickly so that we can move on, but it's from Sizwe. And are there any works in the pipeline for the use of endotoxins produced by the lytic phages, bypassing the specificity of phages to disrupt bacterial cells? Could it be in synergy with antimicrobials and or mentioned strategies by your research group? So I think she's picking up on some of the biocontrol interventions you mentioned.
Yes, so Prof Khan is really interested in the bacteriophage research and the Bordetella vibrio research, all the biocontrol, the biosurfactants and everything. So one of the issues that we have found is you have a problem with bacteriophage resistance developing, depending on how you set up your treatment intervention using bacteriophages. So there are ways in which we can bypass this. But one of the things that we are looking at, which was not focused in this presentation, is how we can use the products produced by bacteriophages or Bordetella vibrio as a way to then also negate the inefficiencies we see with using the Bordetella on its own, or the bacteriophage on its own. So it is definitely something that we are looking at.
Okay, all right. All right, thank you for that. I think so. If you have any more questions for Dr. Reinecke, you can please just either direct message him. He's going to be on the call, I guess, Brandon, and he will be able to respond to your questions. Thank you so much for that presentation. So it's 9:33, moving on to our presentations for this session, and this session 4 now is human health. So our first presentation is from A. Sishuba, and the topic is biodiversity and antimicrobial activity for endophytic fungi isolated from native Sunderlandia fruitus. And our presenter is from Northwest University. So please, when you're ready, you can go ahead. Okay. Okay.
Good day, everyone. My name is Anati Sishuba, and I am from the Northwest University, and I am supervised by Prof. Adeba, and Dr. Mangani. And today I'll be presenting on the topic biodiversity and antimicrobial activity of endophytic fungi isolated from native Pseudolenia fruticans, commonly known as cancer bush. Now we've heard that antimicrobial resistance is currently a problem and is rendering our antibiotics as ineffective, and most of our antibiotics ineffective, if not all. And that is the problem. Now that means that we are faced with trying to come up with a solution in how to actually combat that problem. And this study is actually focused on trying to achieve all of that by trying to see the bioactive compounds that are produced by plants, because we know that plants have been used traditionally to heal for a lot of years. So hence we chose to go about this study. Plants are still considered as one of the most important source of biologically active compounds. Medicinal plants in specific have been utilized globally for traditional healing and have been studied extensively for their pharmacological properties. Medicinal plants are also reported to host fungi that are involved in the co-production of these active metabolites. And these fungi may have also contribute in the biological activity that is exhibited by these plants. Now, endophytic fungi are ubiquitous, symbiotic, sometimes slightly parasitic microorganisms that live within the plant's tissue for all, if not part of their life cycle. They carry out resistant mechanisms to protect the host plant from pathogenic invasion by producing secondary metabolites that have antagonistic activities. Now, with these, endophytic fungi are considered to be a reservoir of active metabolites that can be used to lead off drug development. And antimicrobial resistance has been and still is a major issue to the healthcare system globally. Studies have shown that microbes have developed resistance to antibiotics through various molecular mechanisms. And this is a global problem that has led to an increase in the research featuring endophytic fungi, particularly those from medicinal plant, as a source of new antibiotics. In the current study, our plant of interest is Pseudolenia fruticans, which is commonly known as cancer bush, and it's indigenous to South Africa. Now, this plant gained more attention or attraction due to its capabilities to promote well-being. Cancer bush has been used by different ethnic groups from Xhosas, Zulus, Khoisan, and as a remedy for different infirmities from tribal illnesses such as fever, cold to cancer, HIV, liver problems, and so on. This plant is known to be rich in source of phytochemicals, which have been found to have antimicrobial properties. Now, the aim of this study is to evaluate the biodiversity and antimicrobial activities of endophytic fungi isolated from Pseudolenia fruticans. Now, in order to achieve all of this, certain methods had to be put in place in trying to isolate the endophytes, to check the morphological and molecular identification of these endophytes. And that was done through microscopic identification, DNA extraction, PCR, gel electrophoresis sequencing, and computing a phylogenetic tree, for the isolates. And after that, we had to then move further into now studying or examining the antimicrobial activity that is produced by these endophytic fungi that were isolated from the plant. And that included extracting the secondary metabolites and not just using the endophytes as they are. And also then we had to move into doing antibacterial activity, in which we used disk diffusion assay and agar plug assay, and then we moved on into searching for antifungal activity, in which we use the dual culture method assay, and then we also use the filtrate culture method. And we also had to further go into checking the stress tolerance of these endophytic fungi in order to be sure that, to try and see that, are they only active at certain conditions, will they even be able to withstand other different types of conditions. Now, in isolation of the endophytic fungi, fungal strains were successfully isolated and identified using surface sterilization and morphology techniques. A total of 51 endophytic fungi were isolated from the leaf fragments and giving a percentage colonization rate of 20.4%. After that, we had to now further move into properly identifying these endophytes. And that was done by morphological characterization. The isolates were successfully identified into genus level using morphological characterization. And these are some of the pictures we got from microscope viewing. And that also is a picture of the microscopic characters of endophytic fungi on the PDA plates. And then after that, we did molecular identification, in which DNA extraction was done, PCR was done for the isolates, and then sequencing was also done. And this is a representation of a picture of the gel that was run for the PCR amplicons. And then this is a genetic diversity of the endophytes that were finally identified from the plant. And we ended up with 51 of the isolates, were successfully identified. And these endophytic fungi were confirmed, they were confirmed by PCR, and 17 genera in total were classified into 17 genera. The predominant genuses were Penicillin, Alternaria, Mucor, and Coniocheta, which were identified to species level based on their identities in GenBank. A phylogenetic tree was also constructed. A phylogenetic tree was also constructed in order to show the similarities between our isolates and the grouping and the evolution. And in the end, we ended up with 4 main clusters and 6 subclusters of all these strains. After successfully identifying the isolates that we were working with, we then moved to antimicrobial activities, and we did our antibacterial activities, which was disk diffusion assay and agar plug assay. Now these were done by testing the isolates that we have against pathogenic bacteria, human pathogenic bacteria in specific, where we wanted to now see that if we say that the plant is able to produce these endophytes, and these endophytes are actually able to act, to have the same bioactive compounds as the plant, are they now able to at least show activities when they are tested against pathogenic bacteria? And this was done amongst the few bacteria that were used. It was Listeria, E. coli, Enterococcus, and Salmonella. The fungal extract displayed activity against the bacteria, against one or more of the bacteria that were tested. And 16 fungal extract displayed, managed to inhibit the growth of Salmonella, which if you could look at the graph on our left, we have 2 graphs, the DISC assay and the agar plug assay. We have, for the DISC assay, we did have the most sensitive bacteria, which happened to be Salmonella. And then we were testing 10 bacteria, and one of them was the most sensitive, which was Salmonella, and 6 showed intermediate activity, and while 3 showed resistance. And then in plug assay, 5 showed intermediate activity, and the other showed resistant, and none of them were sensitive to the assay. And the largest zone of inhibition observed was against Listeria, which was 18.3, followed by Menhenium with 16.3, and then they were both inhibited by penicillin. And then 26 endophytic fungi produced a bioactive compound that exhibited antimicrobial activity against at least one tested microorganism. And for agar plug assay, only 2% did in agar plug assay manage to inhibit the bacteria that were tested, and 8 endophytic fungi managed to inhibit the growth of 4 bacteria, and the largest zone of inhibition was 21, followed by another which was 20.6. After testing for the antibacterial activity, we then went further into testing for the antifungal activity. Now, for antifungal activity, plant pathogenic fungi were used to test, because not only is resistance experienced in human pathogens only, but they also, in terms of environmental microorganisms, they're also exhibiting some resistance. And so we used plant pathogenic fungi to test our antifungal activity. And the first assay was done using dual culture assay, in which 84% of the tested endophytic fungi exhibited inhibition against one or more of the tested pathogens. And in this activity, in this assay, mucor, penicillin, and aspergillus exhibited a broad antifungal activity spectrum for 3 pathogens, for 3 or all pathogens. In this case, 4 pathogens were used to conduct this activity. For diversity of antifungal activity, 28% of the endophytes gave a high activity, while 22 gave a moderate activity, and 39 gave slight activity, and 11% of the endophytes had no activity at all. After using this, we had to then move on to another assay in order to just not rely on only one assay to conclude our results, and then that was filtrate culture assay. And in filtrate culture assay, we were using the secondary metabolites of the endophytes. In dual assay, we were using the endophyte as it is from the plate. And then in filtrate culture assay, we were using the secondary metabolites extracted from the endophyte. And in this activity, 87% of the endophytes showed inhibition against one or more of the pathogens. And endophytic fungi, 11 exhibited a broad range activity against all pathogens. The filtrates were able to show activity against the pathogens in varying levels, from most active to most activity at 18%, and active ones at 18%, moderate activity at 18%, and 22%, and slight activity at 9%, and least activity at 23%. Now we noticed that there is antimicrobial activity of some sort from the endophytes that we were using. But now we also wanted to further understand if will these endophytes be able to tolerate different stress conditions? Because we know that fungal communities are responsible for various things in the ecosystem. But the functioning of these fungal species and the functioning of these fungal species is mostly reliant on the conditions in which they're exposed to. So it will be better to further understand if are these active because of a certain condition that they were given, or will they still be active if exposed to different conditions. And during that, we tested temperature, salt, and pH. For temperature, for the test done for temperature tolerance, we used different temperatures, which was 2 degrees, 25 degrees and 37 degrees. And none of the isolates were able to grow at 2 degrees. However, 25 degrees, which is known to be the proper temperature for growing fungal isolates, they all grew. While in 37, some of the isolates grew and some of them failed to grow, as you can see in my graph there, that we do have growth in. We do have some growth which showed very high activities, and we also have high activities, and we also have moderate activities, as well as others failed to grow at that temperature. But now we were able to see that, okay, they can actually move from not only growing at 25 degrees, but also at a higher temperature like 37 degrees. After that, we tested also for salt tolerance. And salt tolerance, we had our media supplemented with NaCl at 3%, at 10%, and at 50%. And the fungal isolate showed extreme growth at the temperature, at the concentration of 3%, where 90% of the endophytes endophytic fungi isolates showed growth, and the concentration was increased to 10% and 84. Now it was dropping. 84 of the isolates showed growth, and when we were at 50, 76 of the isolates showed growth, which is 76 is not that bad. It means that even at 50, some of the isolates or some of the endophytic fungi were still able to grow. The last test that was conducted to test for stress tolerance was pH tolerance, in which we did pH of 2, 5, and 12, and most of the isolates grew in the pH of 5, and the percentage was 44%, followed by 35% for pH 12 and 21% for pH 2. And this supports that some fungi are able to grow in both acidic and alkaline conditions. And in conclusion to this, to the best of our knowledge, the study is to investigate the biodiversity and antimicrobial activity of endophytic fungi isolated from this plant is actually the first study. And also that endophytic fungi isolated from medicinal plant in South Africa do possess excellent antifungal properties against pathogenic fungi and also antibacterial properties. Endophytic fungi are untapped territories filled with effective novel bioactive compounds to control fungal pathogens responsible for mortality rate and high crop loss. And these endophytic fungi have the potential to be used in the natural biocontrol agent in the agricultural sector, antifungal agents in the medicinal, as well as pharmaceutical industries. And okay, these are my references. And I would like to acknowledge my supervisors, Prof. Adeba and Dr. Mangani, NRF for the funding and also the school funding. Thank you so much.
Thank you, Anati, for that presentation. We will take questions that came out of your presentation during the facilitated Q&A. Okay, so we're going to move on to the next presenter now. And the next presenter is going to present on the evaluation of efflux pump mediated disinfectant resistance in bacteria from the University of Free State. So please, when you're ready, you can go ahead and share your screen.
Am I visible and audible?
You're audible, but I don't think I can see you unless I'm not seeing everyone. Yes, Gunther, we can see you.
All right, thank you so much.
Please go ahead.
All right, good morning, ladies and gentlemen. My name is Gunther Staats, and today I will be discussing my project entitled The Evaluation of Efflux Pump Mediated Disinfectant Resistance in Bacteria. So I'm under the supervision of Prof. Robert Bragg and the co-supervision of Dr. Charlotte Boucher van Jaarsveld at the University of the Free State. So the contents of my presentation will include a brief introduction, the project aims, the chapter 2 and 3 results and discussion, some conclusions and acknowledgements. So antibiotic resistance has rapidly emerged and is causing breaches in biosecurity. And oftentimes these breaches are a result of multi-drug resistant bacteria that can be the main cause of the world leading into a post-antibiotic era. So the multi-drug resistance profile is not limited to only antibiotic resistance, but also disinfectant, antiseptic, and heavy metal resistance. So currently, disinfectants and proper biosecurity could be the last line of defense to prevent the world leading into a post-antibiotic era. So disinfectants are currently used in a widespread fashion, being used in a variety of sectors, including the animal husbandry, medical, industrial, and household sectors. And within each, they are used at varying concentrations, and thus understanding how resistance is able to spread between these sectors will be important to combat the threat of multi-drug resistance. So co-resistance is when antibiotic and disinfectant resistance occurs simultaneously in a bacterium or a bacterial population. And this is a cause for concern to combat multi-drug resistance. And oftentimes a key resistance mechanism in co-resistant populations is noted as efflux pumps. So efflux pumps are transmembrane proteins that function to reduce the intracellular concentration of a specific substrate for which they have specificity and extrude it either into the external environment or the periplasm. So here we can see a range of efflux pump families, such as the R&D and the ABC, which are able to form tripartite structures that span the entire membrane of the bacterial cell in gram-negative bacterium and are able to extrude from the cytoplasm directly into the extracellular environment. Other families, such as the MFS, the MATE, or the SMR families, form monomeric structures and are only able to export their substrates into the periplasm. So these substrates are not restricted to only toxic chemicals, such as antimicrobials or waste products, but they also include metabolic or regulatory products or substrates. So an unknown Sriracha isolate was found to be isolated in a DDAC or didecyl dimethyl ammonium chloride based disinfectant bottle in or at the University of the Free State. So Sriracha is noted as an important nosocomial pathogen, especially as a result of urinary tract infections and catheter-associated bacteremia. So uncovering how this Sriracha isolate was able to resist the disinfection within the bottle itself can add to the limited knowledge which is currently disinfectant resistance. So the aims of the project were to examine the efflux pump profile of 2 Sriracha strains using a bioinformatic approach, and to elucidate and verify the efflux pump activity of 2 Sriracha strains or Sriracha strains against or using efflux pump inhibitors. So chapter 2. So the whole genome sequences of the 2 Sriracha strains, namely the HRI, which was isolated in the disinfectant bottle, and ATCC Sriracha marcescens, was accessed on NCBI, and their taxonomic IDs were used to input organism-specific data into the transporter or transport DB program. Now, this program utilizes a relational database to predict the cytoplasmic membrane transport protein complement for organisms whose genomic data is accessible. So, excuse me, this program then classifies the transporters or transporter proteins of each organism into protein families according to the transporter classification system, and additionally also provides a substrate or function prediction. So once the transporters had been classified, each entry was individually curated, and those that denoted any form of resistance, being antibiotic, disinfectant, heavy metal, or multi-drug resistance, as can be seen in the table, were curated and examined or checked using 3 basic alignment tools, namely the KEGG pathway blast, the NCBI blast, and the UniProt blast, in order to confirm the protein identity of each transporter. So this graph depicts the total transporters that were predicted in both Sriracha isolates. And here we can see that the total number in the type ATCC isolate was 733, and the HRI resistant isolate was 796 total transporters. And a large majority of these transporters were identified or predicted to be from the ATP binding cassette superfamily, as well as the major facilitator superfamily. Now, it is important to note that many of the ATP or ABC superfamily transporters are involved in regulatory functioning, as well as metabolic transport. So the next graphic depicts the comparison of the predicted multi-drug efflux systems in the 2 strains. And here we can see the 2 families or transporter families that are most prevalent are the major facilitator superfamily once more, as well as the resistance, nodulation, and cell division superfamily. And these 2 have been noted in literature to be involved in multi-drug resistant profiles, being found in food production as well as healthcare settings. These have also been associated with some bacterial species such as Acinetobacter baumannii, Listeria monocytogenes, Escherichia coli, and Pseudomonas aeruginosa. It is important here to note, however, that there are fewer multi-drug efflux systems noted in the resistant isolate than in the type isolate, being 33 and 37 respectively. So this table just depicts the full multi-drug transporter profile of the resistant isolate, and particular attention can be paid to the highlighted transporter known as MRE, which is identified as the only different transporter having multi-drug functioning compared to the type isolate. Now, MRE is noted to be found in Escherichia coli and has been noted in literature to have the ability to transport quaternary compounds, ethidium compounds, and tetracycline. So the question remains now, does one efflux transporter, would this be sufficient to provide a differing resistance phenotype between the 2 Sriracha strains? The next table depicts the resistance transporters of the HRI strain that did not denote multi-drug resistance. So we can see some antibiotic resistance transporters as well as heavy metal transporters, but importantly, only 2 specific quaternary ammonium compound transporters, and these were SUG-E and QACE. Now, SUG-E was found in the type isolate to also be present, and it is noted in literature that this transporter is found in Enterobacter cloaca. However, knockout mutations of this specific gene showed no increases in susceptibility towards benzalkonium chloride, a first-generation quaternary ammonium compound. QACE, however, was absent in the type isolate, and it is well known to be a plasmid-borne transporter, often associated with staphylococci. But could then this EMRE transporter and the QACE transporter be sufficient, again, to provide the differing resistance profiles seen in the 2 Sriracha strains? So the conclusions of this chapter were that the total identified transporters in the Sriracha HRI were greater than that of the type strain. However, the multi-drug transporters of the Sriracha HRI were fewer than that of the type strain. So because of the novelty of disinfectant resistance currently, many efflux pumps that could be involved in resistance have either been incorrectly identified or predicted, or have not yet been predicted or classified to be involved in disinfectant resistance specifically. Moving on to chapter 3. Just a brief run of the methods. Broth macrodilutions were prepared for the MIC assays for DDAC. 0.5 McFarlane bacterial inoculum was used. A 20-minute contact time was also used. And then each dilution was diluted into fresh BHI or TSA, plated out on TSA. And this graph depicts the results received, where the blue graph was the type ATCC strain and the red graph depicts the HRI strain. So we can see statistically significant differences in the result, or the MIC result for DDAC specifically, which is a 4th generation QAC, between the two strains, suggesting that there is indeed resistance phenotype differences between the two strains. However, the question remains, what is promoting or conferring that resistance to the HRI strain? So efflux pump screening was conducted, where similar fashion, a broth macrodilution was prepared. The efflux pump inhibitor was added to each dilution. A 20-minute contact time was allowed to expire, and the results were streaked out on TSA plates. So this table depicts the results. In the first column, we have the MIC result without the addition of the efflux pump inhibitor. And the second column shows the MIC with the addition of the efflux pump inhibitor, which is reserpine in this instance. And we can see for the HRI resistant isolate, there is a roughly 250-fold reduction in MIC when the reserpine is added to the MIC assay. And for the type strain, we can see there is a 2 to 3-fold reduction in the MIC when reserpine is added. So we can deduce then that there is indeed reserpine-specific efflux pumps present in both these isolates. However, they are more prevalent in the HRI strain. So the MIC assays were then repeated using a 0.5 McFarlane bacterial inoculum in order to compare to clinical breakpoint standards. And again, a 20-minute contact time, dilution into BHI, as well as streaking out onto TSA. So this graph depicts the results. In the blue, we have the standard DDAC MIC for the Sriracha ATCC. The red graph depicts the Sriracha HRI with just DDAC, and the green graph depicts the Sriracha HRI with DDAC and reserpine. So we can see statistically significant differences between the MIC with DDAC and with the efflux pump inhibitor reserpine added, suggesting that there are indeed reserpine-specific efflux pumps present in the HRI strain conferring DDAC resistance, and it reduces the resistance almost to the same level as the type isolate when reserpine is added. So we can conclude then that the screening did show that there are efflux pumps present and that there are MIC differences with and without the efflux pump inhibitor addition. So reserpine is a competitive inhibitor for the substrate binding site of specific efflux pumps, namely the MFS and the SMR family. And we can deduce then that that these efflux pumps confer DDAC resistance to the Sriracha HRI specifically. So efflux pumps could potentially be the fastest and most effective resistance mechanism in the bacterial response repertoire. However, it is unclear in these specific isolates or the HRI isolate if efflux pumps are the only resistance mechanism, as there could be others that are functioning simultaneously over an extended period of time. So better understanding how these bacteria can resist disinfection protocols will add to the limited knowledge, which is disinfectant resistance currently. I would just like to acknowledge my supervisors, Ms. McCarley, the Veterinary Biotechnology Group of the University of the Free State, as well as the National Research Foundation for their funding. Thank you very much.
Thank you, Gunther, for that presentation. Any questions will be reserved for the Q&A that is coming up. And also, in case you just don't want to forget your question, you can also type it in the chat. And when all the presenters are done, I will be able to go through the chat and see what questions were put in there. Okay, so our next presentation then is by Hendrik Stein, and the presentation is on the evaluation of potential novel antibiofilm drugs, also from the University of Free State. So Hendrik, when you're ready, you can please go ahead.
Wonderful. Good morning, ladies and gentlemen. My name is Hendrik Stein, and my presentation will be focusing on the evaluation of potential novel antibiofilm drugs. My study leader is Professor Paul Albertijn, and my co-study leaders are Dr. Hiscock and Professor Albertijn. So with the discovery of penicillin, the use of antibiotics has revolutionized the approach to a treatment of diseases of various kinds. Also, antibiotics have been introduced into different industrialized sectors, including agricultural and veterinary medicine. Each of these sectors relies on antimicrobials, whether it be antibacterial or antifungal, for their success. And the widespread use of antibiotics, however, has resulted in antibiotic resistance. So furthermore, the reckless administration and mismanagement of the currently available antimicrobials have resulted in the establishment of multi-drug resistant organisms and pathogens. And 6 examples of pathogens with growing multi-drug resistance are the so-called escape pathogens. Now, these pathogens have established various mechanisms and properties such as antimicrobial inactivation, bacterial target site modification, reduced antibiotic accumulation, and the persistence through biofilm formation to achieve antimicrobial resistance. Now, when considering the prevalence of different types of resistance in bacteria, it is expected that antimicrobial resistance will become evident in fungal organisms and opportunistic pathogens, such as Candida albicans, for example. The reckless use of antibacterial compounds has established a niche of susceptible hosts with impaired immunological defenses. And these immunocompromised hosts allow for opportunistic fungal pathogens to establish superficial and systemic infections. Now, one of the mechanisms by which microbes can be more resistant to antimicrobials is through the formation of biofilms. In general, biofilm formation starts with the attachment of individual planktonic cells to a surface, and once successfully attached, the cells start to form microcolonies, which grow and produce an extracellular biofilm matrix consisting of extracellular DNA, polysaccharides, and proteins. Finally, a mature biofilm is then produced and cellular dispersion occurs. Now, importantly, the extracellular biofilm matrix is essential to biofilm formation as it serves as the structural support system to the biofilm. It also facilitates cell attachment, intercellular communications, and antimicrobial protection. And all these aspects make biofilms much more resilient than their planktonic constituents. So as an example of 2 biofilm-forming organisms, I will focus on the bacterium Pseudomonas aeruginosa and the yeast Candida albicans. Pseudomonas aeruginosa is an opportunistic pathogen known for its ability to form extensive biofilm structures, as can be seen here in Figure A. And these biofilms are clinically relevant and have been isolated and identified on various medical apparatus, such as catheters and disinfectant soap dispensers. This discovery has emphasized the impact of Pseudomonas aeruginosa in nosocomial infections and resultant deaths. So in contrast, Candida albicans is a commensal yeast found in the human microbiota. However, as an opportunistic pathogen, Candida albicans is responsible for superficial to chronic and deadly systemic candidiasis. The success of Candida albicans lies in its virulence factors that overcome and accommodate various niches in host organisms. The most prominent virulence factors of Candida albicans include its polymorphic nature, its environmental response mechanisms, but most importantly, its ability to form biofilms, as can be seen here in Figure B. Now, in addition to these 2 organisms forming extensive biofilms individually, significant research has been done to investigate when Pseudomonas aeruginosa and Candida albicans form a polymicrobial biofilm in which these 2 organisms have a complex and multifaceted relationship. So much like other biofilms, a polymicrobial biofilm is a continuous interaction between its partners, the host organism, and the environment. And the metabolic exchange between the micro and macro environments define the biofilm's formation, structure, and pathogenic potential. And the polymicrobial interaction between Pseudomonas aeruginosa and Candida albicans is a testament to that. This interaction has been described as antagonistic, as Pseudomonas aeruginosa attaches to Candida albicans hyphae, which results in cell death. However, complementary metabolic responses, promoted quorum-sensing molecule production, and advanced phenotypic plasticity avails a multifaceted system in which antimicrobial resistant properties are utilized. So the arsenal of antimicrobial resistance mechanisms has prompted the immediate efforts to discover novel and alternatives to antimicrobial compounds. As expressed by Nicola et al. Depicted in this diagram, alternative antimicrobial avenues include various options. And of interest to our research group specifically is the discovery of novel antimicrobial compounds. It is evident from the extensive antimicrobial mechanisms provided by a biofilm that there is an immediate call for alternatives to currently available antimicrobials, but more importantly, alternative treatment options and avenues need to be devised to accommodate and include these antibiofilm components. So the aim of my project is to determine the efficacy of novel supramolecular self-associating compounds against mono and polymicrobial biofilms in vitro. Now depicted here is the basic structure of these SSAs. And here we can see various opportunities for substitutions to design the most effective antimicrobial compound. So I just want to draw your attention to the 15 compounds that we use. Just in the interest of time, I won't be going into too much detail, but I would like to draw your attention to compounds 1, 3, 8, 30. We have compounds 5, 6, 16, 33. We have compounds 3, 8, 39, and 60. And then lastly, we have coumarin and coumarin-related compounds, which have either been encapsulated or bound to coumarin itself. So when determining the efficacy of these novel SSAs against mono and polymicrobial biofilms, the following methods and techniques were used. We started by screening the provided SSAs for biofilm inhibition and mature or preformed biofilm inhibition. And to screen the SSAs, a twofold serial dilution was done, and then the optical density measurements were quantified. So Pseudomonas aeruginosa was cultivated and inoculated with the diluted compound in a 96-well plate, and then optical density measurements were taken before and after inoculation to determine the efficacy of this compound. Similarly, for Candida albicans, the organism was cultivated and then again inoculated with the diluted compound in a 96-well plate, and optical density measurements were taken before and after inoculation to determine the efficacy of the compound. Finally, the same methodology was applied in the case of the polymicrobial biofilms. However, the standardization step did differ. And then again, we inoculated the compound, the diluted compound, taking optical density measurements before and after inoculation. So just some preliminary data. What we found looking at the biofilm formation, specifically just the normal SSA structures. When looking at Pseudomonas aeruginosa, we can see the compound 5 worked the best at an MIC of 2.56 millimolar at approximately 20% percentage reduction. With respect to Candida albicans, we can see the compound 39 worked the best at an MIC of 2.56 at approximately 45% reduction. When looking at the polymicrobial biofilm, interestingly enough, we can see that compound 30 worked the best at approximately 45% reduction. However, compound 39 was also very effective at 40% reduction. We can relate this compound 39 activity to the previous set of results indicated here, suggesting that maybe in the polymicrobial biofilm, the compound worked better on the Candida albicans as opposed to the Pseudomonas. So what we did then is we took these SSAs and we did some contact time inhibition screening, and we found that compound 5 showed a lot less activity in this regard. However, we did see some not dose dependent, but rather time dependent responses with some of the compounds. And again, compound 6 just here showing some activity, but not enough to be significant. With respect to Candida albicans, however, we could see that compound 33 showed a wonderful activity here at approximately 35% at the 30-minute contact time period, as well as 10-minute contact time period. And then when looking at the polymicrobial biofilm, we can see that compound 5, again, with some of the other compounds, it does seem that there is a time-dependent response, where interestingly enough, compound 39 did not have any activity whatsoever. So just in summary, when we have a look at the data, it seems that compound 6 and 33 work the best when looking at the data set. However, more experiments and tests will be done just to make sure everything is reproducible. So now looking at the coumarin and coumarin-related compounds, just testing on biofilm formation, we can see that coumarin had a dose-dependent response here at 2.56 millimolar, having a wonderful percentage reduction. We can see that compound number 70 had a fairly good activity throughout the board at a MIC of 0.16 being the last that we see any activity. With respect to Candida albicans, we can see that the coumarin was not effective at all. However, compound 71 had a wonderful activity at approximately 95% at an MIC value of 2.56. For the polymicrobial, we can see that coumarin has now turned. It has been very effective, again, elucidating to the fact that it might just be reacting to the Candida albicans in the polymicrobial biofilm as opposed to the Pseudomonas aeruginosa. And finally, all the other compounds do seem to have good percentage reduction activity. Now, in the second data set, again, for the coumarin-related compounds, we can see the same tendency when looking at the coumarin, again, with good activity. We can see that the other compounds still, except for compound number 70, here at an MIC value of 2.56, had percentage reduction activities of interest. Looking at the Candida albicans here, we can see that coumarin, again, has wonderful percentage reduction activity, as well as the other compounds. And finally, with regards to the polymicrobial biofilm, we can now see that coumarin again has been effective against all compounds, and especially compound 62, which has been shown to be effective across board. So just to compare data sets 1 and 2, we can see the data set 2 has been the most effective. Compound 63, interestingly enough, could not be tested in data set 1, which means that we would have to reproduce the data and just perform the experiments again. But it would appear that the SSA coumarin-related compounds definitely show some activity, or inhibitory activity rather. So just to conclude, it would appear that the following compounds of the 15 tested, compounds 6, 33, 62, 70, 63, and 71 are the most effective at inhibiting biofilm formation and preformed biofilms. And this has led us now to devise the toxicity screening, which will be done next on the model organism Caenorhabditis elegans. But we look forward to presenting that data in the near future. I would like to a moment just to acknowledge and thank the following. Thank you very much.
Thank you, Hendrik, for that presentation. Likewise, please do keep any questions that you might have for Hendrik in the chat box, please, and we will answer those during the Q&A. So then our last presentation for this session is by Motsepi Plachi, and the presentation is going to look at medicinal plants as therapeutic agents for trypanosomosis, a systematic review, and the presenter is from Northwest University. Motsepi, when you're ready, please go ahead. Yeah, we can see you. We can't hear you.
Sorry.
Okay.
Are we on?
Yeah, perfect. You can go ahead.
Good day. My name is Motsepi Plachi, and I'll be presenting a systematic review on medicinal plants as therapeutic agents for trypanosomosis. Trypanosomosis, sorry. I don't know what to do. Sorry, I'm struggling a bit. I can't see my screen. Caused by pathogenic trypanosoma parasites infecting both human and livestock. The disease is known as sleeping sickness or human African trypanosomosis in humans. The livestock disease is called Nagana or African animal and Odemia. There are other two more animal trypanosomosis syndromes, which are called Dorine and Sura, which are caused by Trypanosoma equipedum in equines and Trypanosoma evansi in various domestic animals, respectively. According to the World Health Organization, human African trypanosomosis affects between 300,000 and 500,000 people worldwide, with an estimated $4.5 billion in annual economic losses due to animal trypanosomosis. The major strategy to control trypanosomosis relies on the use of trypanocidal drugs, which are challenged by an increasing problem of resistance. The obstacles emphasizes the need for research of alternative new compounds and effective. That's why we use plants, because plants are potential sources of new drugs due to the presence of countless number of secondary molecules that have pharmacological effects. The aim of the study was to document the findings from several independent studies on the in vitro and the in vivo efficacy of medicinal plant extracts against and humans. materials and methods. For search and strategy and study selection, all scientific English publications published between 1990 and November 2020 were retrieved from PubMed, ScienceDirect, Scopus, SpringerLink, and Web of Science database. And then the following keywords combination were used: medicinal plants and anti-trypanosomal activity. The relevant papers were first screened by their title and abstract. One reviewer used predetermined eligibility criteria for screening, and then the data was confirmed by another investigator. We also assessed the risk of bias. This that we use for this systematic review. We used it for both in vitro studies and in vivo studies. So for in vitro studies, we used a checklist called the Guidance Document on Good In Vitro Method Practices. And for in vivo studies, we used the ARRIVE guideline. And then for results, Figure 1 shows the Prisma flowchart depicting the main steps in the process of selecting eligible publications. So we identified 851 through the mentioned database, and then from those 851 records, 251 were excluded because there were duplication. And then we were left with 600, and then from those 600, we excluded 386 studies based on title and abstract, and then we also excluded 57. And then we were left with 157 full text articles that met the eligible criteria. And then from there on, we excluded 84 when we looked at the criteria. And then for this systematic review, we only used 69 articles. From those 69 articles, 53 were in vitro studies, 10 were in vivo studies, and then 6 conducted both in vitro and in vivo studies. And then from those studies, most of them, they were conducted in Nigeria. And then Caya senegalensis was the most investigated plant species. And then Fabaceae was the dominant plant species. Only 19 studies reported on synthetic compounds or fractions or oils, and then only 12 studies reported phytochemical screening on crude extracts. And then figure 3 shows the summary of bias analysis on the in vitro and the in vivo studies using plant extracts for the treatment of trypanosomosis. So in A, which is the in vivo studies, no action was taken to reduce the possibilities of errors, for example, mix up to avoid cross contamination. And then about 90% of the studies, the materials and method were well written and documented. And then about 58% of the studies met the test definition, which includes the purpose and the scientific basis of the test. And then in vitro studies, 46% of them reported ethics committee permission. And then all of the studies included animal species, doses, administration route, and the experimental design. In discussion, more studies were found to evaluate in vitro antitrypanocidal activity of medicinal plants as compared to in vivo studies. Advantages over in vivo test, including being less time consuming. And then the dominance of Fabaceae family may be because it includes herbs, shrubs, and trees. And then since only 23% of the studies included a thorough phytochemical screening of the plant, to the discovery of particular compounds with biotechnological potential for use in the development of novel medicine, and then only studies using extract function and isolated molecules were able to overcome this limitation. In conclusion, there are promising medicinal plant extracts that are potentially effective in the treatment of trypanosomosis, and among these plants are Caya senegalensis, Zimmeria americana, and Mytinus senegalensis were the most widely studied plant species. This systematic review assisted in identifying medicinal plants for future research focused in the development of safe, affordable, and easily accessible plants for the treatment of trypanosomosis. And in order to minimize the risk of publication bias, basic guidelines or protocols must be included for experimental investigation in both in vitro and in vivo studies. And these are some of the references that I used, and I would like to acknowledge the Northwest University and NRF for funding, and thank you for listening.
Okay, thank you so much, Mtsepi, for that presentation on your systematic review that you conducted. I think at this time, we are going to take some questions. There are some questions that have already come through on the chat. So if our presenters are ready, the first one goes to Anati. And Anati, Hazel is asking, what is the significance of salt in endophytes, and why was it only mineral tested?
Sorry, can you repeat that question, please?
So Hazel is asking, what is the significance of salt? Remember, you showed us some results about salt tolerance.
Okay.
So she wants, yes, so she wants to know what the significance of that is. And then why was it only mineral tested?
The significance of salt, well, it serves as nutrients. Yeah. It serves as nutrients to the microorganism.
Okay, and so what are the implications of that then from a perspective of making sure that the endophytes can kill off whatever organism you're testing it against? So maybe in terms of halophilic organisms, maybe you could expand on that.
I'm sorry, I don't understand.
Okay, all right. I wanted you to relate it back to the application of the work that you're doing so that it could give more context to the question that Hazel is asking, because I think she probably doesn't know about the different types of organisms in terms of halophilic organisms, and how if your endophytes are able to have a high salt tolerance, then it shows you that they can survive in that kind of environment with those same type of microorganisms that might be present as well.
Oh, okay.
Yeah.
Well, we were testing for that so that we could honestly understand, try and understand if those endophytes were able to tolerate, because some studies actually just show that certain endophyte of fungi, they're only able to tolerate a certain level of salt tolerance. Now we were trying to up the dose in order to understand if will they be able to still grow if the salt tolerance is a bit higher.
Okay, all right. And then there's also another comment. I'm not sure who it's from, but it says, I think, Anati, this is also obviously to you because you were looking at some of the indigenous occurring endophytes. She says, not only are you promoting the further studies of indigenous knowledge systems, but also antimicrobial awareness. Why must we do research for extracting antibiotic compounds from endophytic microbes? Isn't it enough to extract antibiotics directly from plants? Is it assumed that the endophytes are reservoirs of antimicrobial compounds found in plants, or or are standalone microbes. So I think it's just the explanation of the relationship between endophytes and the antimicrobial compounds that you're getting from there. And isn't there an easier route to go to obtain these compounds that we need?
Oh, okay. Well, in terms of that, it is known that the endophytes actually produce the same bioactive compounds as the plant. So yes, we can isolate from, we can extract from the plant, but now there are limitations in terms of using only the plant, because now with extracting from using endophytes gives you, there are limitations of using the plant alone, because the plant grows seasonally, and it can be a lot of work in terms of extracting in the lab, or it can give small amounts of what is this, of the compound we are looking for. Take, for instance, if I were to now work with leaves directly as they are, let's say one leaf will only amount to a certain gram of compound. But now if we extract the endophytes and we actually work with the endophytes instead, that gives us an unlimited, but it gives us a big range of in terms of extracting. Plants, they can only give us a limited amount of antibiotics. Imagine if we were to extract maybe 100 grams of antimicrobial compounds from one plant. It's biomass. It's biomass because you have to crush it and all of that. You may end up to maybe 10 or 30 grams of the antibiotic. But in microbes, they are self-replicating and can be kept at a closed environment, and they may be able to even produce more. Like in fermentation of alcohol, the mass is just, it gets bigger.
Yeah. So basically it's about the biotechnological applications of these endophytes. So like you said, once you're able to have the endophytes, then you can now produce it in mass quantities, compared to always going back to your plant where you're probably even going to get losses as well. Okay. Thank you, Anati, for that, and also thank you to the attendees for those two questions. I don't see any other question for the other presenters, but for Ganta, I have a question. So you talked about this disinfectant resistance that you're now seeing. So what kind of chemicals then can we use in order to sort of maintain the bacterial sensitivity and obviously not to generate more efflux pump resistance? Because I mean, obviously with COVID, we've been using tons of disinfectants, and I can imagine the kind of resistance that's going to come up either from an efflux pump point of view or from something else. So what kind of chemicals then can we use to kind of combat that?
Thank you very much for your question. I think from literature, it was a lot of times they suggest that rotational use of disinfectants that have differing modes of action would be the most effective way to reduce the resistance that can occur or can be promoted in bacterial populations. But like you said, with the results of the COVID pandemic, many of the hand sanitizers that are indeed sold are mainly ethanol based, and how that ethanol resistance is going to, or the increased ethanol being exposed to your dermal bacterial populations on your hands and all the environments that or high touch surfaces will also impact the resistance genes that are being induced in those populations as well, and how that affects other disinfectants like quaternary ammonium compounds is still to be seen. But yeah, I think the best method would be rotational use of disinfectants currently.
Okay, thank you for that. Any other questions for Okay, so just as I was going to say any other questions, one question has come through, and it's from, I think from Dr. Sam. She is asking, of the medicinal plants identified to treat sleeping sickness, which ones do we have here in South Africa? So Motsepe there.
Hello.
Hi. Did you hear the question?
Yes, I heard the question.
Okay.
In South Africa, we haven't done medicinal plants on trypanosomosis as yet. Because even when I was doing the systematic review, like South Africa did not appear in any of the databases. So yeah, it shows that we do have to do this research here.
Okay, and yesterday we actually had a presentation by, I think it's Dr. Betek.
Betek, yeah.
Yes, he was looking at some synthesis, I think, of some molecules, and I think one of them, antiparasitic diseases, he did mention, sorry, one of the parasitic diseases he did mention was trypanosomosis. So maybe it would be, I'm not sure whether he's your supervisor or you have some kind of research relationship there, but it would be also good to see what he's investigating, because he presented on some work around that organism.
Oh yes, I remember his presentation. Yeah, maybe I need to collaborate with him and look into it.
Okay, great.
Yes, thank you.
You're welcome. And I think that is it so far. And I also think that I will round off the session now so that it will give us at least a 5-minute break, because session 5 will be starting at 10:45. So once again, I would like to first thank our keynote for this morning, Dr. Reineke, setting the scene for us in terms of the overall presentations that we actually heard. So a lot of the presentations touched on some of the things he also talked about in his main research work that he's conducting at Stellenbosch University. And then I'd also like to thank our 4 presenters this morning, Anati, Gantha, Hendrik, and Motsepe. Those are very interesting presentations. And we look forward to some of how the work progresses, since some of the work is still in the early stages. And also thank you to our listeners and those who send through questions also for the participation. So we will continue at 10:45. Thank you.
Right. Good morning, everyone. My name is Nonhlanhla Kalabaila, and I am from the Water Research Commission. Welcome to this session, which is session 5, which has a theme on environmental health. So today we are very fortunate. We have a keynote address from Professor T.J. Barnard from the University of Johannesburg. And then after that, we have 4 more presentations for different topics on environmental health. And then we are going to take a short break and then come back and then have 2 more presentations under the theme Global Recovery. Right, so the very first person to speak to us under session 5 is Professor Tobias Barnard from the University of Johannesburg. So Professor Barnard is the director of the Water and Health Research Unit at the University of Johannesburg. And then he's also part of the WRC community of researchers. He has done a lot of work on microbiology in general, but also a lot of work on antimicrobial resistance. Prof, over to you, whenever you're ready.
Good morning, everybody. And like Nonhlanhla said, I'm part of the furniture. I think I started quite early or late with the water sector, but it's been fun being part of the water sector. And I thought I'd take a lesson from Dr. Barnes yesterday and just sort of speak from my perspective on antimicrobial resistance and how I got into it. I'm not a trained, or I was never planning to go into the water sector. I was trained as a biochemist, and more likely, or more precisely, I was a gene jockey that was looking at expressing funny proteins in bacteria for commercial products. And that is what I thought would form the basis of my presentation today. The simple question or assumption: we are safe. Plasmid transformations only done in the laboratory. The reason for this is that in last year's talk, I spoke about what we're doing with antibiotics in the laboratory and are we disposing of it in a safe manner. And this year I thought, let's talk about the bacteria and the genes. And of course, we have to we have to focus on one bacteria, and my bacteria of choice will always be, so it's not going on. Apologies. My organism of choice will always be E. coli. So you'll see a funny slide. I wanted this as a t-shirt. I wanted this as a screensaver. The simple reason I was in love with E. coli. I used E. coli for all my experiments in my master's and my doctorate and my honors. I was using E. coli to clone just about anything into a plasmid and putting it into E. coli. At that stage, we never thought really about the impact on the impact of antimicrobial resistance, because we use plasmids with a variety of antimicrobial genes, marker genes that we used. And the more complex the cloning, the more complex the antibiotics, the more interesting the antibiotic combinations we use in the plasmids. But we use it to express proteins for a more biotic approach. We would then put these plasmids into E. coli. And like I said, my favorite organisms, there's a joke that goes, E. coli is such a wonderful bacteria because you can put just about any DNA into it and it will accept it. Something that has become a problem for us now. So the joke is now on us. We would then take the plasmid, put it into the E. coli and screen for the presence, or screen for colonies with the plasmid using the antibiotic resistant gene, as well as then in my case, an activity. So in my PhD, I was looking for lipases, so I was looking for a zone of clearance. We would use these colonies and we would start growing them up into more and more larger volumes. At one stage, we were growing up our organisms in 25-liter bioreactors just to make enough protein or enough of the cells to get enough of the lipase produced, so we can start doing the characterization of the lipase. And this sort of brought us always to the first conundrum, or the first question we got asked for using the genetic modification of E. coli. But this is dangerous, and you're looking at putting weird genes into E. coli. What if it spreads into the environment? Number one, thankfully, we weren't putting in dangerous genes into the E. coli, but we would always say that transformation of E. coli, or simply putting in a plasmid into an E. coli gene, is not so easy. We were all taught in our microbiology, biotechnology, biochemistry classes that you have to have one of 4 or 5 processes using chemicals, physical or electroporation or something to put a gene or a plasmid into E. coli. And if you've done enough experiments in the laboratory, you know that the cloning and transformation process itself can be a very tiring and frustrating process. So we never thought that it was going to be a problem. And from that perspective, we would always go, no, what we're putting into the DNA is safe. Don't worry about it too much. It's not going to influence it. We were all taught about antimicrobial resistance, antibiotic resistance, but we never thought about the real impact of this because we were mostly working in the laboratory, not thinking about the environmental sciences or the clinical sciences. But again, we never thought of a plasmid being carried into the bacteria because we killed our bacteria before we discarded of it. We had this huge, nice autoclave. Everything was autoclaved. So the bacteria was dead. Why was everybody stressing about? I'm not going to go into more detail about this because I've had the privilege of keynote speakers and presenters dealing with this before me, and we're going to hear about this more afterwards. So I'll leave the fun details to them. So coming back to this whole slide of how we produce the enzymes, we would always be left with a huge pile of waste. We would take the bacterial, the E. coli cells, we would lyse it, we would use the supernatant, and we'll start cleaning out the enzymes, leaving the DNA with the lipids and everything around, and that was, well, I like to think always autoclaved and then discarded, but we generate a lot of waste. And I was but one of a few students in the laboratory doing this. And I wasn't even doing full-time bioreactor work. So this is something that really stuck to me. Now, the first question he was going, but okay, but this is biotechnology. Like we said, there's no real danger, et cetera, et cetera. But then the people looking at antimicrobial resistance started playing with these genes. We as scientists want to understand things. The only way to understand things is to study it. And the most effective way to study it is to start tampering with the genes. We start tampering with the folding of the protein to see if we can't stop it. We start putting it into different orientations on the genomes. We start studying these things. I mean, this is just one example of an article where they played with colistin resistance. So now all of a sudden, what biotechnologists have been doing is coming closer to the water game. We now have E. coli with resistance genes or resistant plasmids carrying the colistin resistance gene. So it's coming closer to home. But remember now, we're not only looking at the genes itself, we have to look at what else is out there. I didn't want to embarrass people, so I thought, let's just take my postdocs articles. This is not self-promotion, although there's some small part of self-promotion, but think of what we have been doing in these 2 articles. We went looking for pathogenic bacteria. We isolated the pathogenic bacteria. We then looked for the antimicrobial resistant bacteria. We grew these up in unnaturally high volumes. We studied them, we stored them, and we have to discard the waste somehow. Again, we always felt happy that I'm autoclaving, there's a waste disposal protocol, so everything is being put away. These are but 2 examples of what we're doing. Look at the program that for today and yesterday, and you'll see the types of organisms that we're dealing with that's going through the same process continuously. And you have to remember, my first take-home message is, keeping all this in mind, you always have to remember that bacteria want to survive. As much as we want to get rid of them, bacteria want to survive. So when we start playing within the laboratory, they are going to use what we are testing to their advantage. I'm not sure who of you have seen this video, and if you haven't seen this video, go look at it and watch it on YouTube, where the Harvard Medical School took this nice mega petri dish, mega plate, and they grew this, they poured the plate starting from 0 antibiotic up to 1,000 in the middle, 1,000 times the exposure E. coli would have to have for this antibiotic. So normally we would use 1 time, so they went 10, 100, and 1,000 times. This picture shows you how the bacteria started adapting. So this is the mutational lineages that occurred up to where the E. coli grew at 1,000 times the exposed normal exposure limit. And this happened in 11 days. Think of how long you play with your bacteria in the laboratory. In 11 days, this E. coli could adapt to 1,000 times a normal concentration. So if we are playing in the laboratory with these things, you can only think of what they're doing. And I know I don't have slides about this, but you have to think of E. coli or bacteria as really trying to adapt. And they are vicious in their adaptations. Go read up on articles about suicide genes, where E. coli, for instance, when you're growing your bacteria or your E. coli on petri dishes with antibiotic discs, will create a small wall between them and the antibiotics by sacrificing bacteria. They would go into the suicide genes would be activated, and they would literally kill themselves off to protect the bacteria behind them from the antibiotics, plus give food for the other bacteria to survive, allowing the bacteria behind them to start adapting to the antibiotics being tested. So you always have to think of, if I'm doing Kirby-Bauer discs, how am I disposing of the media? How am I disposing of the organisms? And it's important because we will soon be responsible for what's happening. Now, a lot of this has been coming, we're generating a lot of waste. We're generating a lot of waste in both in water, solid waste, etc. And we have to ask ourselves, what is happening to our waste? Up to about, let's say, 2 days ago, I was happy with the thought that my waste is autoclaved, so I'm happy. Researchers have shown last year that if we're looking at laboratory wastewater, where we have antibiotic-resistant bacteria and antibiotic-resistant genes, simple autoclave, following a normal autoclave protocol, was effective in reducing the antimicrobial bacteria, almost removing all of them, but was not effective in removing the antibiotic-resistant genes, which means these genes were still being released into the environment. They found that only when combining autoclaving with ozonation could you get a 5 to 7 log reduction in the antimicrobial resistant genes, eliminating them from the environment. I know my laboratory is not doing this, and I know that some of the wastewater treatment plants aren't doing this. So are we really treating our waste properly? We then have to consider the other parts of the waste, and I'm just giving some examples. I'll put this into context for you now. If you look at the middle article, where we're looking at solid wastes, researchers have started going to dumps, waste dumps, to see what is going on with these, the bacteria that's being dumped there. Remember, you have some of the sewage being dumped there as well, some of the sewage byproducts being dumped there, laboratory waste, medical laboratory waste, everything is being dumped there. They started noticing that the antibiotic-resistant genes are being shared amongst the organisms. And if you do air sampling on these spots, they isolated bacteria that had carried up to 16 antibiotic-resistant genes that was not supposed to be there. So our wastefuls is becoming a problem for when we're doing this. I borrowed these two articles from some of the previous keynotes, and I got to thinking, if we're looking at these genes and the antimicrobial resistant genes in the water, how much of this is from the environment, from other disciplines, and how much is this from our waste that we are generating? This led us to when we're stumbling across this whole concept of spillover and antibiotic resistance. And I think this was the basis for when we started looking at the nexuses, how they influence, as well as the One Health approach, where we need to start looking at how can what we're doing, or how does the one sector influence the other sector in the or influence the other sectors. But we as scientists tend to look at this from one perspective and from one side alone. What is happening? What is our contribution to this? What are the scientists putting into the system that could be contributing it? Are we contributing, are we producing the superbugs and giving the bacteria all the tools they need to be able to do what they want to do in the environment? Now, coming to the topic of we are safe plasmid transformation done only in the lab, up to date, we showed that bacteria could be killed. We were happy with that. We now show that antibiotic resistant genes couldn't be removed, which is kind of freaky and scary, especially if you have complex systems like the wastewater treatment plants. But we could still go, oh, but the bacteria was at least dead. So the DNA was inaccessible, or the DNA was free around in the environment, so the E. coli can't do anything with this. Unfortunately, we have now been shown that this is not the truth. More and more articles are coming out where they're showing that horizontal plasmid accumulation through transformation in E. coli cells. An article published recently, I isolated an environmental E. coli strain that is naturally competent and can acquire exogenous DNA. The graph I'm showing you is just an example of, this is the environmental strain compared to the normal laboratory transformation strain, where they were put into different types of water, wastewater spills, etc. I think this was molecule water, and the bacteria was still able to take up the DNA. Now it's starting to create a problem for us. We no longer have the excuse, but at least the cells are dead. If we are putting the genes out there, the bacteria is getting used to it, and they're trying to take this up to survive. Because remember, take home message one: bacteria want to survive, and they will do anything to survive. Coming back to my favorite poster, and I'm still going to get this made as a t-shirt. One day you'll see me at the next conference. Look at one of the headings: 60 new antibiotic resistance for this summer. I thought it was funny the first time I saw this, and I thought, isn't it interesting that how funny scientists can sometimes be? Until we started looking at some E. coli that we isolated 3 months ago as part of the project with Professor Vassal and her group. I think Dr. Benin presented some of this work earlier, where we have E. coli that was isolated from normal water samples in Johannesburg. And this isolate, WRC18F, can now wear this t-shirt because it has antibiotic resistance to 16 antibiotics. So no longer are the genes or the jokes funny, it's becoming a reality, and it's getting closer and closer to home. How long until this whole block is purple and we don't have antibiotic options, or antibiotic resistance that these bacteria don't have anymore? Coming to the end of my presentation, you've got to remember that bacteria, like I said, want to survive and are successful at doing this, and they've been doing this for a long, long time. Now that we are able to access the Antarctic soils with the melting of the ice, they are isolating bacteria from what would have been pristine areas with known antibiotic resistant genes. So these bacteria have had these. Now we're introducing more and more bacteria that already have the genes into our environment. And the same will happen to anything that any of the presenters are saying. If it's occurring in a plant, if it's occurring naturally, there's most likely an organism that's already resistant to it or knows how to deal with it. And we've got to keep an eye on that. And that takes me to my second take-home message is, you make sure you're aware of how you work with your bacteria and your bacterial DNA. We do not want to be the contributors to the next outbreak or the next lineage of super resistant bacteria, because these bacteria are there and they're just looking for that little nudge or the tool to go above and beyond the rest. Now, all this work is easy to present, or it's fun to present, but can't be done without a lot of help. And I have to thank the people in my laboratory, that's the core people, postdocs and staff that is doing all this work. We have to finish thank the National Research Foundation for funding students to student bursaries. I have to do UJ Pete's. for helping us with some of our projects. Two of the photo shy people that's not here, Dr. Heine and Dr. Deleuve, that's been doing the work, as well as Professor Vassal Khan and her group from the Water Research Laboratory in the University of Stellenbosch. And then lastly, just in case you didn't notice the UJ branding, I have to thank UJ. I have to thank the UJ Research Committee that's making our research so easy to do with all the work they're doing. And I'm not sure I'm supposed to show this after the previous presenters, but please remember to wash your hands and disinfect your hands so we can stop the spread of COVID. Thank you very much.
Wow, thank you very much, Prof, for that very sobering presentation. And I mean, from some of the things that you've already spoken about, I mean, in my mind, I'm already thinking that I think something that we haven't really thought about is, I mean, we've always said that waste or wastewater serves as a reservoir for resistant genes. But I think what we've never actually thought further to say that then if it is a reservoir, how does it contribute into now the dissemination into the environment in general, not just the aquatic environment? I heard that you also spoke about how the resistant genes can even spread into air. So, I mean, in my mind, that is something that really maybe, as the WRC, is something that maybe we can start looking at seriously, and as well, if maybe the University of Johannesburg it is something that they would want to really pursue and go into, the WRC would really be looking forward to collaborate on that. All right, let me not hog the space. I don't see any questions other than everyone echoing the sentiments of how great the presentation was. I will keep checking the chat for questions if there are any that I directed for you. But at this point in time, I would like to thank you very much for that very insightful presentation. And then I will then, when it's time for the discussion, and then maybe call you back if there is that question for you. Thank you very much.
Thank you.
Right, we are going to move along now. And like I said, we have about 4 presentations today that are on environmental health in general. And then please remember, as the presenters are doing the presentations, please do drop your question if you do have any. And it will also be useful if you can just indicate as well who the question is directed to. And then after that, we are going to pause a bit to check if there are any questions. And then after that, we are going to then move on to 2 other presentations under the theme Global Recovery. Right, without then wasting so much time, this morning we are very fortunate. We have some perspectives from outside our borders. So the very first presentation is by Iwa Kotlarska from the Institute of Oceanology, Polish Academy of Sciences. Right, over to you, Iwa. Right.
Good morning, everyone. I hope you are seeing my presentation and hearing me well. So my name is Ewa Kotlarska. I'm from Poland, from Institute of Oceanology, Polish Academy of Sciences. I think that you are wondering why I'm here. I previously worked with Professor Carlos Bezeidenhout, so I was able to join you to talk about Poland and the Baltic Sea. So today I will talk about potentially pathogenic and drug-resistant bacteria in the Gulf of Gdańsk in the Baltic Sea, Poland. First of all, I would like to talk about my institute for a minute. It's located in the coast of Gulf of Gdańsk. Here you can see the pictures and the picture from outside my building, actually, taken early in the morning. And we conduct research on the Baltic and Arctic seas. We have a research vessel, and we have several departments: chemistry, biochemistry, genetics, ecology, and physics departments. So we are interdisciplinary in marine research. Baltic Sea is the world's largest brackish water environment with limited water exchange. Average salinity is 7.5 PSU, and it's nearly 5 million people living in the catchment area. And we have 11 cities with more than 500,000 citizens in the catchment area, and almost 15 million people live within 10 kilometers of the coastline. And the Gulf of Gdańsk is part of the Baltic Sea, where my institute is located in Sopot. On the map, you can see larger and smaller towns, harbors and marinas, larger and smaller wastewater treatment plants, rivers, also shipwrecks and dredged material dumping sites, and also various dumping sites from Second World War, dumping sites of munitions. So these point sources of pollution are causing that nutrients and other substances are slowly diluted in the Gulf of Gdańsk. So the Gulf of Gdańsk is heavily impacted anthropogenically. So because of that locations, we picked up 5 sampling points, two largest wastewater treatment plants in Gdańsk and Gdynia, 2 marine outflows, which are sites in the Gulf of Gdańsk in which wastewaters are discharged, and also a mouth of the Vistula River. Vistula River is the second largest river in the Baltic Sea catchment area. As you can see, we checked the presence of E. coli, Enterococci, and Pseudomonas, and you see the great reduction of wastewater of these bacteria in wastewaters, in marine waters. In marine waters and river estuary, we have the number of bacteria below 100 CFU per 100 milliliters. This is according to the new bathing water directive. This quality is better. This water quality is better than excellent, but the reduction of bacteria through the wastewater treatment is, as you can see, very good. Excellent, in fact, but through wastewater treatment survive the worst bacteria. I mean, antibiotic resistant with antibiotic resistant genes, even with multi-drug resistance phenotypes. We used several methods to research those bacteria. I will show you today only the examples of that. So we taken wastewater and marine water and marine sand samples, and we are. Sorry for that. We plated the samples using serial dilutions or membrane filtration techniques on various media, and then isolates were identified and their antimicrobial susceptibility was observed. We did that with Phoenix automated microbiology system, and then we obtain breakpoints, antimicrobial breakpoints according to the UCAST recommendations, and then all the isolates were subjected to molecular analysis with PCR screening for antibiotic-resistant genes and integrons, and also for sequencing. Here you can see one example of the of our studies. We obtained almost 600 isolates from fecal coliform agar and from wastewaters and from marine waters, and most of them are E. coli, but we also obtained Citrobacter, Klebsiella pneumoniae, Shigella, Plesiomonas, Aeromonas, Pseudomonas, and even Vibrio cholerae. Here you can see antibiotic resistant phenotypes obtained for E. coli. Mostly isolates from E. coli were resistant to amoxicillin, tazobactam, sulfametoxazole, fluoroquinolones, and also we checked the presence of integrons in those isolates. As you can see, presence of integrons was positive, presence of resistant phenotypes was positively correlated with presence of integrons, and also presence of integrons was correlated strongly with multi-drug resistant phenotypes. Multi-drug resistant phenotypes, I mean that those strains were resistant to 3 classes of antibiotics. Also, those resistant phenotypes in E. coli from wastewaters and marine waters mimic the usage trends in Europe for treating E. coli infections. We also checked the presence of resistant genes in E. coli isolates. As you can see in this table, we checked the presence of genes for resistance to beta-lactams, quinolones, but only plasmid-mediated resistance to quinolones, resistance to aminoglycosides, tetracycline, and sulfonamides. As you can see, mostly we detected gene BLA-TM from resistant to beta-lactams, SUL1 and SUL2, and TET-A. Moving on to Vibrio cholerae, it was quite a surprise because Baltic Sea waters are temperate waters, so we didn't detect Vibrio cholerae before, but when we isolated those strains, the temperature of water was above 20 Celsius degrees, and fortunately, the Vibrio cholerae isolates were non-O1, non-O139, and they did not produce cholera toxin, but they were resistant to antibiotics, and all isolates also possess integrons. Moving on to Pseudomonas, we detected several species of Pseudomonas in wastewater and marine water samples. Pathogenic Pseudomonas aeruginosa was 18% of all strains, but dominant species was Pseudomonas putida, but also Pseudomonas fluorescens and pseudoalkaligenes were also present. Most of the strains were resistant to ceftazidime, aztreonam, T-carcinin, and T-carcinin with clavulanic acid. Pseudomonas pudida was often with multi-drug resistant phenotype. It means that those strains were resistant to all tested antibiotics except colistin. And also very important note that Pseudomonas putida resistant to beta-lactams and to fluoroquinolones and multi-drug resistance were more prevalent after wastewater treatment, but the resistance rate decreased in marine water samples. Moving on to Aeromonas, we detected several species of Aeromonas, Sobria, Veronica, Caviar, Hydrophila, Salmonicida, and group of Hydrophila and Caviar. Recently, we detected Aeromonas media strain with integron 3, which is very rare in the clinics and very rare in the environment. This strain was subjected to NGS sequencing, and data analysis is in progress. But I can say that this strain has two plasmids. One is INSQ with integron on it, and the second one is from INSP incompatibility group. Among those Aeromonas strains, we detected 17 isolates with ESBL phenotype. All ESBL resistant strains were multi-drug resistant and possess class 1 integron, and half of them had BLA-CTM3 gene. This gene belongs to the group 1 of CTXM family of beta-lactamases. As you can see in the global distribution of those genotypes, in Poland, you can see M3 is prevalent in the clinics, so there is no surprise that those strains are present in wastewaters and finally in the marine waters. So to conclude, our studies show that wastewater treatment plants had a good efficiency in preventing marine waters from fecal contamination. However, there is a positive selection of antibiotic-resistant bacteria, antibiotic-resistant genes, multi-drug-resistant phenotype, and mobile genetic elements during wastewater treatment processes. We detected presence of human pathogens in marine waters like Klebsiella pneumoniae, Pseudomonas aeruginosa, Vibrio cholerae, and also Aeromonas, Citrobacter, and Enterobacter genus. Antibiotic-resistant profiles of E. coli correspond to the consumption of antibiotics in medical treatment. Also, we detected presence of antibiotic-resistant genes, including ESBL coding genes and plasmid-mediated quinolone resistance. Also, we detected presence of integrons, and those presence correlate with antibiotic resistance, especially with multi-drug resistant phenotype. Thank you very much for your attention, and I would like to, at the end, acknowledge programs, funding agencies, and people involved in this research. Thank you very much.
Thank you very much, Iwa, for that lovely presentation. And I think the takeaway message from your presentation is that at least wastewater treatment works in Poland, and that also that maybe we also need to follow suit in South Africa to look at maybe the prevalence of antibiotic resistant organisms as well as genes within the marine environment. I mean, which is still related to that previous comment that I said that I think we have just stopped at that wastewater treatment work and aquatic environment interface, that transfer from wastewater into the aquatic environment. But we have not looked further than that to look at further dissemination into the marine environment as well as into the atmosphere. So there aren't any questions for you at the moment, but please stay with us and I will let you know if there are any questions when we get to the Q&A session. Okay. Thank you very much.
Thank you.
Right, our next speaker is Karabo Tsulu from the Noordwes-Universiteit. Karabo, over to you whenever you're ready. Unmute.
Unmute myself. Good morning, everyone.
Sorry, Karabo, just put it on slideshow just before you begin.
Yes. I've been trying to do so. My computer is
Fantastic.
a little bit slow. Yes. Good morning, everyone. My name is Karabo Tsulu, under the supervision of Prof. Carlos and Dr. Lesoho Malalo-Tom. The title of my project is Incident and Characteristic of Antibiotic-Resistant Bacteria in Raw and Drinking Water from Waste and Keep Water Production Facilities. Introduction. The discovery of antibiotics has caused a massive change in medicine by protecting lives against pathogenic bacteria, thus improving life expectancy of humans and animals. According to the South African Bill of Rights, everyone has the right to have access to free medical health care services, and as a result, the government provides free medical health care to 42 million people. There is insufficient data on the consumption of antibiotic resistance. South Africa also has a problem of using antibiotics in agriculture that are banned in countries with public health care facilities. Most banned antibiotics are reserved for humans due to their toxicological reasons and lack of evidence to treat animal-related infections. Another problem that we have in South Africa is that antibiotics that are intended for livestock farming are readily available over the counter. Literature also states that primary healthcare prescribers in South Africa play a significant role in the emergence of antibiotic resistance. A study conducted by Fele and colleagues showed that 97.1% of medical healthcare prescribers in South Africa accept that antibiotics are extensively used. 95.8% believe that antibiotic resistance is an alarming issue. To be honest, I don't understand why these numbers are not 100%. The last finding made by Fele and colleagues is that 66.5% of medical health prescribers feel forced by patients to prescribe antibiotics for them. The improper and extensive usage of antibiotics in veterinary medicine, human medicine, and agriculture has led to the spread of antibiotic residues in wastewater treatment plants. Already, South African wastewater treatment plants are not working effectively. A total of 318 out of 163 wastewater treatment plants were reported to be non-compliant against the Green Drop certificate. This is according to the Department of Water and Sanitation, and this report was published in 2017. Non-compliant wastewater treatment plants creates favorable conditions for microorganism, nutrients, antibiotic-resistant bacteria, antibiotic residues, and antibiotic-resistant genes to be spread into receiving water bodies that may be used for production of drinking water. Literature also states that drinking water production facilities are not designed to eliminate antibiotic-resistant bacteria and antibiotic-resistant genes. Currently, antibiotic-resistant bacteria and antibiotic-resistant genes are not monitored in environmental as well as drinking water, thus making drinking water a perfect vector for antibiotic resistance. Consumption of contaminated water sources pose public health threats to immunocompromised patients, children, and elderly people. The aim of the study was to determine the incidence and characteristic of antibiotic-resistant bacteria in raw and drinking water. The specific objectives were to determine the physical, chemical, and general microbiological quality of raw and drinking water, to isolate and purify heterotrophic plate or HPC bacteria, to determine antibiotic-resistant pattern and the associated gene, to determine whether HP isolates are potential pathogenic, to identify HPC isolate using 16S rRNA gene sequencing. This diagram is a schematic diagram of one of the drinking water production facility that I collected my sample from in the Western Cape. This drinking water production facility is situated in a water-stressed region of the Western Cape, and as a result, it uses or it recycles wastewater to compensate for the high demand of drinking water. This treatment plant uses advanced treatment processes such as ultrafiltration, reverse osmosis, advanced oxidation, and the water that is treated via reverse osmosis is then blended with borehole water and dam water that has underwent conventional treatment processes before it is discharged or distributed to the public. Okay, the second treatment plant that I collected my samples from is named WCF, of which it treats water using just the normal conventional treatment processes. Raw water sample, treated water, and water distribution networks were collected. Physical parameters were measured on site, and chemical parameters were measured upon arrival at the Noordwes-Universiteit laboratory. Microbiological agents were also determined. Heterotrophic plate count bacteria were subjected to gram stain to determine their purity. Only pure isolates were subjected to capillary diffusion method, and antibiotic-resistant isolates were subjected to pathogenicity test. Isolates of antibiotic-resistant isolates and pathogenic isolates was extracted using the ChemMagic kit, and the DAD was subjected to endpoint PCR for amplification of 16S rRNA gene and antibiotic-resistant genes. These genes were sequenced to get the identity of HPC bacteria and antibiotic-resistant genes. Results now. The physical parameters measured at plant WCA were within the South African standards or SANS for drinking water. Similar results were recorded at plant WCF. The chemical parameters measured at plant WCA were also within the South African standards for drinking water. Also, similar results were recorded for plant WCF. For microbiological agents in water, two general trends were observed in which fecal coliform and E. coli were not observed in drinking water, and there was a clear reduction of HPC bacteria from raw and treated water. As anticipated, a lot of HPC bacteria were present in raw water samples. Some bacteria belonging to the genus Bacillus and Pseudomonas were present in both raw and drinking water. It is of great concern to report HPC bacteria in drinking water that were absent in raw water samples. Similar results were recorded at plant WCF, in which bacteria belonging to the genus Bacillus and Chrysobacterium were present in both raw and drinking water. Again, there were some bacteria that were only reported in drinking water that were not present in raw water samples. However, the problems lies beyond physicochemical parameters and microbiological agents into antibiotic resistance. This table is a summary of antibiotic resistant patterns observed for 12 antibiotics. As we can see that isolates display the lowest resistance to cephalothene, chloramphenicol, erythromycin, kanamycin, neomycin, tetracycline, and streptomycin. And isolates displayed the highest resistance to ampicillin, cephalothene, and trimethoprim. As anticipated, isolates with the highest resistance were isolated in raw water samples, but this was not the case at the WCF plant. We saw an increase of resistance from raw into treated water. It is of great concern to report antibiotic-resistant bacteria in recycled water that was intended for drinking purposes after treatment and distribution network. However, amongst the drinking water sources, recycled wastewater had the least antibiotic-resistant isolates. From the Capybara susceptibility test, it is evident that wastewater, raw water and drinking water samples are contaminated with antibiotic-resistant bacteria. Hence, it was important to calculate the multiple antibiotic-resistant indices, or the MI indices. The MI indices is a useful tool to determine whether a sampling site has a low or high use of antibiotics. And water environments that have high antibiotic use have the MI index higher than 0.2. Table 8 and Table 9 indicates that most sampling sites, including drinking water samples, had high antibiotic use. This table represents antibiotic-resistant genes and virulence characteristic isolates at plant WCA. Antibiotic-resistant genes such as ERMF and ERMB that confers resistance to erythromycin belonging to the family macrolides were more prevalent. Other resistant genes include MC and INT1. Some isolates also displayed beta or alpha hemolytic characteristics, and some produced DNase, gelatinase, lipase, chitinase, or proteinase extracellular enzymes. The isolate that displayed the highest virulence was detected after treatment, as it is shown by the red highlighting here. This isolate displayed phenotypic resistance to 11 antibiotics. It harbored 2 antibiotic-resistant genes, and it was beta hemolytic and produced all 4 tested extracellular enzymes. Similar results were obtained from plant WCF, in which ERMB was the most prevalent antibiotic-resistant gene. Again, the isolate that displayed the highest virulence, it was detected after treatment. This isolate displayed phenotypic resistance to 10 antibiotics. It harbored ERMB resistant genes. It displayed beta hemolytic, and it produced all 4 tested extracellular enzymes. This is just a gel. It's just a gel electrophoresis of a 1.5 agarose gel of the 4 successfully amplified antibiotic-resistant genes. In conclusion, the physicochemical parameters and microbiological agents in water were within the drinking water production, the science of drinking water. However, the problem extends beyond the physicochemical parameters and microbiological agents into antibiotic resistance. This study was able to expose gaps in South African literature as far as studying for antibiotic-resistant bacteria and antibiotic-resistant genes in drinking water production facilities. We observed that drinking water production facilities are unable to completely remove antibiotic-resistant bacteria and antibiotic-resistant genes. Hence, it is important to study or constantly monitor antibiotic-resistant bacteria and antibiotic-resistant genes in environmental water and drinking water, because consumption of contaminated water sources may pose public health threats to immunocompromised patients, children, and elderly people. I would like to acknowledge these bodies, and thank you.
All right, thank you very much, Karabo, for that presentation. And just a reminder to our audience, if you have any questions for Karabo, please just drop them into the chat or the Q&A. Right, our next speaker is Dr. Bronwyn-Kibi-Makala from the University of Western Cape. Bronwyn, over to you.
My apologies in advance for the little co-worker. I hope my screen's showing okay. Good morning, I'm Dr. Bronwyn-Kibi-Makala, and I'll be presenting our work which focused on the detection of antibiotic-resistant organisms, as well as the enumeration of ARGs in domestic grey water systems. Come on.
Bronwyn?
Yeah.
Are you sure you'll manage, or would you want me to push your presentation towards the end?
No, he's gonna be here the whole day. So if it's okay, I'm happy to carry on.
No, please do carry on.
So between 2015 to 2018, the Western Cape province experienced the worst drought in 113 years. And there was a real threat of day zero. So that was the day that the taps would run dry. This drought saw the implementation of strict water restrictions coupled with increased water tariffs. Residents were obviously encouraged to adopt innovative approaches to water usage. And the use of grey water was strongly encouraged for activities which did not require fresh water. So for those that don't know, grey water is defined as all household water, which does not have fecal contamination. And grey water collection can range from simple bucket collection to automatic filtration systems. Now, grey water composition can vary widely. It also depends on factors like the household demographics. One thing that is consistently shown is that it has significant chemical and microbial contamination. So which brings me to the problem statement of our projects. So knowing that household grey water may contain residual antibiotics, which coupled with those conditions within the systems themselves would support the proliferation and the growth of antibiotic-resistant organisms. So the aim of our study was to perform phenotypic analysis of strains isolated from the grey water systems, as well as developing a qPCR assay to detect the ARGs linked to resistance to 3 types of antibiotics. So this is just a brief overview of the methodology we used for the entire study. So for the study, we collected 10-litre grey water samples as well as biofilm samples with sterile swabs from 4 households approximately every 3 months for an 18-month period. These samples were analyzed by the combination of molecular and microbiology techniques. So our first aim was to isolate bacteria from the grey water and the biofilms and to study their diversity. So what we found is that the microbial communities were similar across the systems and they remained stable pretty much throughout the testing period. So that was the 18-month period. Microbial abundance was high, which can be seen by those lawns in the undiluted sample, but surprisingly the species diversity was low. And typically we only saw about 4 to 6 morphology types at each sampling site. So initially we decided to perform ADRA to determine how many different species were present in each sample. We decided to just sequence 1 representative clone from each ribotype, and the results just from that initial little screening is put in table 1. We found that obviously most of the species were related to clinical strains. So Klebsiella was found in the biofilms. Enterobacter was found in the grey water, and then Pseudomonas was detected in biofilms, which obviously is interesting because within clinical settings, you often find Pseudomonas species in those clinical biofilms. Samples were then screened for antibiotic resistance using the WAAW method. So from the grey water samples we analyzed, we found that there was commonly resistance to teicoplanin and cefodoxime, with some of the isolates showing resistance to streptomycin. Strains generally showed susceptibility to ampicillin and cethlothin, as can be seen by the zones of inhibition. One thing that we did find is in the biofilms, we often found only 2 colony morphologies, so like these common types of cells, which we called the large and big colonies, and we analyzed their resistance separately. We found that both these organisms were resistant to teicoplanin, which is obviously slightly concerning because this is not a widely used antibiotic. And studies have shown that organisms that are resistant to teicoplanin are also likely to show resistance to vancomycin. And obviously that's, like everyone knows, is a last resort antibiotic. Both of the colony types were also resistant to streptomycin. So for the second part of the phenotypic screening, we assessed the level of antibiotic resistance by using very old-fashioned antibiotic gradient plates. And in order for this part of the study to align with our subsequent qPCR study, we decided to only use the 2 antibiotics, ampicillin and streptomycin. We isolated 22 isolates, which were resistant to both antibiotics, and then we screened them for the resistance. So of the 22, we found that 19 had some level of resistance to ampicillin and 14 had resistance to streptomycin. Only 3 grey water isolates were fully susceptible. So all other isolates of the 22 showed some level of resistance. And alarmingly, overall 64% of the isolates showed resistance to both antibiotics, with ampicillin resistance being particularly high in the biofilms. Selected strains were then selected for 16S sequencing, and we found that the strains were similar to common soil or wastewater, or on the other hand, like clinical isolates. Most of the nearest phylogenetic neighbors were also reported to be multi-drug resistance. For example, isolate B1b at the top of the table in blue had 100% similarity to Chrysobacterium endogenes, which has been reported to be resistant to both ampicillin and streptomycin in clinical settings. Similarly, halfway down the table, we have 4GW and 5GW, and these were found to be most similar to Zingomonas species. Now, Zingomonas spea is often found in grey water, soil, and wastewater. And it has been reported that there are high levels of streptomycin resistant. The prevalence of wastewater organisms in these systems, particularly in the biofilms, would support our hypothesis that these systems are in fact colonized by stable microbial communities. And we're not just detecting organisms which are just transient or non-metabolically active. The presence of clinical strains like the Klebsiella is obviously not unsurprising, as the grey water included water that came from human bathing and hand washing. So for the last part of the study, we used qPCR to actually detect the AR genes. As qPCR results are dependent on having really good quality RNA to start with, we did spend a considerable amount of time optimizing the RNA extraction and the cDNA synthesis. These grey water and biofilm samples were really challenging to work with, as they were riddled with PCR inhibitors, and the amount of starting cell mass was very low. So initially we evaluated 5 extraction methods. So that was a conventional bead beating method, the Trizol method, the hot phenyl SDS, the CTAB, and then the commercial RNase easy kit. So they, obviously by using those kits, we looked at different lysis and precipitation methods. So ultimately, we found that the hot acid phenyl SDS method was the most successful and consistently yielded the most RNA, which also had the highest level of purity. Interestingly, even when we compared it to the commercial kit, which is specifically designed to extract RNA from water samples, the hot phenol method still performed better and resulted in a 100-fold increase in yield and a significant increase in RNA quality. This can obviously be seen from the table and in the gel image on the right. So finally, to ensure that our results would be a true reflection on gene expression and to prove that we are actually detecting RNA transcripts and not DNA, we performed 2 rigorous Ambion turbo DNase-free treatments on all the samples to ensure that there was no genomic DNA contaminating the samples. So from the gel that's in red, it can be shown, from the gel on the left-hand side, it can be seen that even after the RNA was still present, remained intact, and we only had a minimal loss in yield. And then after converting those samples to cDNA, we did a 16S PCR to confirm that there wasn't any DNA present. And the absence of the 1.5 band on the gel confirms that there was no genomic. So only after the samples had been fully processed were they then used in subsequent gene expression studies. For qPCR analysis, we decided to target 3 antibiotic resistance genes, the VAN-A, the AMSEAM, and the AADA. And we selected those due to the relevance in the clinical stressors, particularly infections associated with escape pathogens. And the 3 reference genes we used was 16S, GAP-A, and MDH. So from the MELT curves we obtained for the target genes after the optimization process, it can be seen that we only got a single amplicon, so that confirms that the primers were specific. And from the standard curves, it can be seen that all of the standard curves have an R-squared coefficient value of greater than 0.99, which confirms that the primers were efficient and then up at an acceptable level to use in later analysis. So I'm only showing the preliminary results here. So we just tested the VAN-A primers on selected grey water biofilm samples. So VAN-A gene is part of the gene cluster, which encodes for antibiotic resistance to vancomycin. And although we only think of vancomycin in the clinical setting, many studies have shown that VAN-A is frequently detected in water samples. So from the analysis, we found that we couldn't detect the VAN-A gene in 2 of the 6 samples we initially analyzed. And in the other samples, the levels was about 5.1 to the exponent 2 or 1.4 to the exponent 3. My apologies. And the detection of the VAN-A gene in 4 of the 6 analyzed samples and the fact that we found higher levels in the biofilm is consistent with our phenotypic results, which showed that several of the biofilm isolates were resistant to vancomycin. So just a summary of our results. So phenotypic analysis found that there were viable resistant organisms living in the grey water systems, and about 60% of these isolates were resistant to at least 2 antibiotics. The low species diversity we found is likely linked to the fact that these systems are being improperly used and the water is being stored for longer periods, as it's been shown that diversity decreases during storage time. So preliminary qPCR analysis revealed that antibiotic resistant genes are actively being expressed. And this study highlights that domestic grey water systems are likely a source of ARGs within the environment, and the general public needs to be made aware of the potential health risks, especially if the water is being stored for long periods and then discharged into the environment. And I would just like to acknowledge the following people. And yeah, my apologies for my badly behaved co-worker.
Thank you very much, Dr. Kibi Makala, for that presentation. And yeah, I must say, I admire your multitasking skills very much. I need to come over to the university to learn a thing or two.
Sorry, he was sick today.
Yeah. Okay, but yeah, we had the gist of the presentation. I think we might have to give your co-worker a project in future so that he can also be busy. All right, I am going to be checking the chat if there are any questions that are directed to Dr. Kibi Makala, but thank you very much for that presentation. At this point, we have to move on to the next presentation, which is also from outside our borders, similar to the first one that we had. And this presentation will be delivered by Rolof Kotsie from Gothenburg University. Rolof, over to you. Unmute.
Yeah. Okay, can you hear me?
Yes, yes, I can.
Can you see my presentation?
Yes, just not in slideshow. Ah, perfect.
Perfect. Okay. So my name is Rolof Kotsie. I'm a postdoc at the University of Gothenburg, and today's presentation will be a software that we've developed, GenView, a gene-centric phylogeny-based comparative genomics pipeline for bacterial resistors. Lots of big words. So first of all, who are we? Well, we're the University of Gothenburg, and we are found, if I can just put a little pointer here, we are found over here on the west coast of Sweden, and Stockholm is over here. It's the second biggest city in Sweden. We are part of the Department of Infectious Diseases, so we have lots of clinical annotations, and we work in the hospital. And we are also the instigators of the Center of Antibiotic Resistance Care, who is a multidisciplinary stakeholders collaborative initiative to do research in the field of antibiotic resistance from various departments to work together to solve this global issue. So I just want to mention some notable contributions so that you might recognize who we are and what we do. Recently, we published an updated version of the state of antibiotic resistance in environments in Nature Reviews. This article I will be talking about a bit on later on, about the origins of antibiotic resistance genes. We mentioned in a couple of other papers also some other important aspects, such as the discovery of the SOL4 gene, and also an article that I'll be touching on briefly is the antibiotic resistant genes in the environment, prioritizing the risks that ruffled some feathers about, is it actually a risk to have an antibiotic resistance gene in the environment? So first, a bit of background over here. First of all, the problem 10, 20 years ago in microbiology was simply that we had too little data to do anything. We couldn't make any conclusions on a genetic level. But now current problem in microbiology is that we simply have just too much data to analyze. So it's sort of a weird problem. And this is where the need for bioinformaticians come in. So this is a graph from GenBank showing us the amount of sequences that have been generated over the past 30 years of GenBank. And then also in the red, the number of users. So as you can see, it's taken off since the year 2000 after the Human Genome Project. And again, after 2010, when sequencing technologies came a bit more user-friendly, cheaper, and more readily available. What I would like to point your attentions to is the incorporation of an antimicrobial resistance database in GenBank only 2 years ago, which is a great thing for us, but I mean, we are behind if we look at all the other stuff. And another thing I want to highlight is, if you look at, we've got 6 million users daily on GenBank, and it's important that we utilize this technology to make deductions in our research. And some people might still be stuck over here when we're like sort of using it sometimes, but it's very important that we start using these tools that are available to us to make deductions in our research. So GenView is a specific solution to a very specific problem. It's not your typical pay-to-play type of software because it's very specific, and which should be the way forward for most software, in my opinion. The problem with existing pay-to-play tools is the lack of variability. It's a one sort of directional tool that you can use to do something broad, like metagenome assembly, but you can't ask specific questions. So in today's world, it's very common to analyze antibiotic resistance genes out of context, which still, of course, has its place in research. For example, having to look at diversity, phylogeny, mechanisms of action, and so on and so forth. But what about mechanisms of evolution? We can't really deduce that from analyzing an individual antibiotic resistance gene. All that we can say is that they do occur in the environment and that they are diverse. But But this doesn't really tell us much about the risks, does it? Is it a risk to have a known antibiotic resistance gene in the environment? Probably not. All right, but I'll ruffle some feathers and we can discuss that a bit later. So what's important to look at in the context of evolution is the mobile genetic elements surrounding an antibiotic resistance gene, which tells us more about its capability of disseminating amongst the population, in between species and among species. But what about genetic environments between different species or different sampling locations and so on and so forth? How do we compare these mobile genetic elements in the context of an antibiotic resistance gene between various species? This is where GenView comes in. It's a fully automated gene-centric pipeline, meaning we focus on a single antibiotic resistance gene. It uses parallelized computational steps, so it's very fast. It uses a bunch of bioinformatics tools to make it work. And there's a difference between a software and a tool. A tool is a thing that has a very specific goal, and we utilize these tools to answer a specific question. The input can either be user-defined or from the NCBI database when you insert your sequences. And the plot is interactive. So it consists of two steps. First of all, the user input is given by your own assemblies or from the NCBI database. Then the genes are extracted, the open reading frames are determined, and the predictions are done based on the open reading frames, and annotation is done. The second step is to cluster these different ARGs and environments into clusters, and then creating a phylogenetic tree of those specific environments. And finally, the output is a gene visualization. So I'm going to treat you with a little example of what I'm talking about. This is the output, and I hope this works. So please, let's just go back one. Let's just see if I can open this up for you. So what's cool about this software is the fact that you don't need any fancy type of software to analyze the data. You can use your browser and it's exported in HTML file, which means you can send it to your colleagues. There are different types of features. You can change the opacity of the genomic environments. You can change the scale so that you can sort of zoom in to the different genetic elements. You can reset the scale. You can create an image for publication purposes. You can change the distance from the phylogenetic tree. Also, you can make it a medium or a small version of your genetic environments for better visualization. So that's pretty much how the whole thing works. What's the potential applications of this type of software is to do stuff like determine the origins of antibiotic resistance or antibiotic resistance genes to better predict where it might be going. So in this example from a paper that was published by Stefan Ebmeyer, which is the lead author of this software that we've created, he also used this in an application, and he used it to identify the origin of antibiotic resistance in some species. And how it works is if you, let me just get my pointer back. If you have known genes in a chromosome with specific genetic environmental constructs, and you follow the antibiotic resistance gene to a known position where it is an actual functional ARG, and if you find the same gene on a mobile vector, such as a plasmid, surrounded by mobile genetic elements that was found on the chromosome and its historical variants, then you can make the reasonable deduction that it was originated from that organism. And this is what he's done in this regard to determine recent origins of antibiotic resistance genes where they sort of translocate themselves across taxa, which is worrisome. And of course, it's something that should be kept in mind. From this research, he found that most genes, origins of most antibiotic resistance genes are from the proteobacteria group. But what's also very important here is that only 5% of the origins can be identified. The other 95% are still unknown and probably happen somewhere in the environment. And these various origin routes must be identified, and the software allows this to happen. It's very easy to install using the Conda install bioconda channel. And for those of you who've worked with these types of things before, it will see it works quite nicely. So the conclusions we can draw from this as a whole is that big data is the future cornerstone of all science. With the current amount of 7 million sequences on GenBank, no, I think it's more, on GenBank, that it's impossible to analyze by hand. And certainly you need more bioinformaticians with more types of specific skills to answer specific questions. For example, when we look at, let me just go next one. Right, so we need niche specific software to understand the complexities of biology. So what I mean with that is that when you understand the evolution of genes, it opens doors in context. When we look at big data over here, volume and value, these are pretty much established by laboratory work. But as soon as you come in dealing with, is the information misleading? Can it be visualized? What's the variety, the velocity, the viscosity, and the virality? Then this is where bioinformatics comes in, to put all of this into an analyzed way and to produce a user-reader format. Future applications can lead to estimation of transfer events and management of antibiotic resistance with this software, which is an important aspect in future for management and identification of novel genes and how they might spread throughout the environment. And finally, and I can't stress this enough, open source software. People might say, how do we know your software is accurate? Well, it's open source, which means you can go look at the code yourself. And you know what, if you have a problem with the software, you can change it. And we encourage you to do that. We encourage you to take the software and make it better. And that's what scientific research and collaboration should be about. I would like to acknowledge the University of Gothenburg, the Chalmers University, the funding entities, CARE, the different co-authors. Over here is Stefan, which was the lead author, and this is Joachim Larsen, our PI. And finally, I would like to just remind and invite all of you for the ADAR conference next year in Gothenburg, Sweden, which is hosted by the Gothenburg University. And that is my story. Thank you so much.
Thank you very much for that presentation, Roelof. Very, very, very interesting. I think yours and another presentation that we listened to yesterday from Dr. Pienaf, it just reinforces the need for more bioinformaticists so that we can be able to make or extract value from the data that we have so far. And also the need for data analysis or data analytics or new tools for maybe extracting more value and having more information from data that we would have already have. All right, that brings us to just the end of those 4 presentations that were scheduled for this very first session, a sub-session, and session 5. So we have 2 more presentations with the theme Global Recovery. At this point in time, this session was allocated now to taking some few questions, if there are any, on all the 4 presentations and even the keynote from Professor Paranad that we had at the beginning of session 5. I do not see any specific question in my chat, so I am just going to pause maybe just for a few minutes, just in case people are still gathering their thoughts around, just to field a few questions maybe to. Okay. Okay, please do. Please do, go ahead.
Right, so the first part, can you use whole genomes? How by identifying a specific loci, which is a specific antibiotic resistance gene, and then it uses upstream 10,000 base pairs and downstream 10,000 base pairs to identify which environmental or other genetic factors might be associated with that specific gene. So whole genomes, no, I think whole genomes is practical. And my mind, I'm thinking, why would you want to do that? Because surely there can be different associations across the genome that might have an influence. But in this specific scenario, we're looking at the immediate overlapping environment. The second question was, sorry, what was the second question? Own database, I think it was. Yeah. Can you create your own database with plasmids? And sorry. Yes. So there is an option in the software to use exclusively plasmids. So yes, you can definitely create your own database in plasmids. Yes, absolutely. And the third, sorry, what was the third question? Limitations right now, I would say, they probably are. It hasn't been used extensively enough that we could have identified. I think a specific limitation might be when we have unknown gene regions, hypothetical genes, and so on, that might they identify them in the context of the genetic environment, but we still don't know what they are. So it's sort of redundant. They might as well not even be there. But it's still worth going to look at what they might be, which is something that I found is that often we have genetic environments with lots of hypothetical genes, and it's only the loci in the middle. And it's sort of like, okay, what does this tell us? Not really much. So that might be a limitation of the software, but that's something that certainly can be can be improved. But what the advantage in this regard is that it predicts open reading frames, which sort of tries to alleviate this shortcoming. I hope that answers the questions.
I hope so too. Thank you very much for those questions. I don't see any other questions. Thank you very much, Rudolf, for the presentation and also for the comments that you've just given now. Then that brings us to the end of this session. The next sub-session on global recovery will resume at 12:20. So that gives us about 7 minutes or so as a break. And then when we come back at 12:20, we are going to listen to two presentations, one from Refilwe Mabeyo, and then the next one from Denzel Leiki, those two presentations which have the theme global recovery. Right, see you shortly at 12:20. All right, welcome back to the next subsession on global recovery. Under this subsession, there are only two presentations. The first one will be by Refilio Mabeo, and then the second one will be by Denzel Leakey. So without further ado, I am going to give this opportunity to Refilio to start with the first presentation. Refilio, are you ready? Right. Please switch on your camera as well as your microphone.
Okay. Can you see me? Can you hear me and see me?
I can hear you. I can see you. Please go ahead.
Okay. Hi, everyone. My name is Refilio Mabeo, and I will be telling you about the escape pathogens in wastewater treatment plants into receiving waters excellence program under the supervision of Dr. Luciho Tom, as well as Prof. Paulus and Dr. Didi van Zeek. I will not be sharing my video because I have internet difficulties. Okay, so in 2007, the World Health Organization developed a global priority list of 12 bacteria against which new antibiotics are urgently needed due to multi-drug resistance. Now, the most critical group of all includes multi-drug resistant bacteria that pose a particular threat in hospitals, nursing homes, and among patients whose care requires devices such as ventilators and etc. And this includes Acinetobacter, Pseudomonas, as well as various members of the Enterobacteriaceae family. These bacteria have become resistant to a large number of antibiotics, including carbapenems and third-generation cephalosporins, of which at present there are the best available antibiotics treating multi-drug resistant bacteria. Now, in the priority list, they were categorized into critical and high of new antibiotics that are desperately needed. And under the critical, they listed an Acinetobacter baumannii, Pseudomonas aeruginosa, as well as members of Enterobacteriaceae. And under priority 2, which is high, they included Enterococcus faecium as well as Staphylococcus aureus. Escape pathogens. The escape group consists of Enterococcus, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, as well as Enterobacter, as they are listed here in this picture. And they have been reported to develop multi-drug resistance and extensive drug resistance in many developing countries. This group of microorganisms is problematic since the research has revealed that they are associated with the highest risk of mortality, thereby increasing healthcare costs and antibiotic costs and usage. The commonly found to be problematic are vancomycin-resistant Enterococcus, methicillin-resistant Staphylococcus aureus, vancomycin-resistant Staphylococcus aureus, penem-resistant Klebsiella, as well as ESBL-producing Klebsiella pneumonitis, to name a few. All of the above are causing serious health outbreaks and challenges in neonatal units, with fatalities reported in some of the cases. There is a considerable database on the surveillance, research, and development aspects of escape pathogens in the clinical setting, particularly with respect to the hospital-acquired infections caused by these species. Since these microorganisms since these pathogens are known to withstand and escape the action of antibiotics and other disinfectants, research has suggested them as possible indicators for antibiotic resistance in the environment. The environment as a potential source and reservoir of pathogens has for a long time been ignored. According to WHO, the spread of antibiotic resistance occurs through interactions between animals, plants, and people in the shared environment. Now, wastewater treatment plants are regarded as hotspots as they are somehow middlemen between waste coming from various sewage sources and the environment. Accumulation of waste from various sources turns wastewater treatment plants into unintentional collection points for antimicrobial agents, ARVs, as well as ARGs. Wastewater treatment plants are not designed and not yet upgraded to remove ARVs and ARGs, and as a result, they are known to have antimicrobials and other agents associated with antibiotic resistance. Due to this challenge faced by wastewater treatment plants, high concentrations of antibiotics and their associates in the wastewater treatment plants effluents are released into the environment via wastewater treatment plants discharge to rivers, wastewater reuse, and irrigation. Now, the aim of this study as a whole is to evaluate the prevalence and characteristics of escape pathogens in the wastewater environment and use this data to develop a surveillance program. And this is going to be achieved by the following objectives, which is quantify ARGs and virulence genes and escape pathogens isolated from wastewater, compare the levels of escape pathogens using cultivation and qPCR methods based on eDNA in samples collected from influent and effluent of selected plants, compare the whole genome sequences of escape isolates to sequence from clinical isolates in public databases, and lastly, to use this information from the three objectives to design a surveillance program for monitoring escape pathogens in selected wastewater treatment plants. So for this particular presentation, we'll only be focusing on the surveillance program. For now, we have two wastewater plants that we are working with within the Northwest province, and seeing figure 1 shows the wastewater treatment plant in the neighboring town, and it's called a biofiltration wastewater treatment plant. And on the second figure, we have the biological treatment plant that we have here in Potchefstroom that we are working with. Due to time, I will not be going through the processes that go into this plant. Surveillance program. What is the surveillance program? It can be defined as the core tool in understanding the nature and scope of the problem while assisting in controlling the problem at hand. Surveillance programs comprises of compiling, analyzing, and disseminating data on the problem posed, in this case, the presence, characteristics, and prevalence of escape pathogens in the environment. Effective surveillance is significant in both understanding and controlling the spread of the problem. It also allows assessment as well as evaluation of possible intervention and implement plans. Here we have a proposed surveillance program plan that we will be working with. And firstly, we start with establishing goals, and these are your research gaps. This is basically identifying the problem at hand. And secondly, design a comprehensive surveillance system. Now, this involves, since we're working with wastewater treatment plants, this involves going to the plant and getting the setup of the plans to see what kind of processes that are already there, and trying to see where the problem could be potentially coming from. And from there onwards, we go to select appropriate personnel. This involves having a person that you will be in contact with at the plant, that will be monitoring what's coming in and what's coming out on a day-to-day basis, since we cannot do that by ourselves. And then from there onwards, we acquire tools and variance analysis. This involves now looking back into our results and discussing on possible solutions that can be implemented on these plans according to the results and findings that we have. And lastly, implementation of the system. Now this will involve talking to relevant stakeholders, the government, and try to raise this concern to them, since they have all the findings, and try to show them the importance of why these plans need to be implemented. The various programs set up in the ESCAPE project, and in-depth analysis of escape pathogens from the environment, as well as the characteristics, will provide information on their incidence, prevalence, potential sources, as well as pathways. This information will be critical when intervention monitoring programs are set up and implemented on local, regional, and national scales. And the latter programs will, in the long run, assist in optimizing empiric and targeted antibiotic prescription choices. Despite numerous studies in the clinical setting having investigated escape pathogens, minimal data is available regarding their presence in the environment in South Africa. The WHO also stressed that limited, accurate, and reliable data are limited, and as a result, the true full extent of the problem is unknown. The significance and benefits of establishing a surveillance program for escape pathogens. This study will be of importance as data generated can add great contributions to the whole global resistance surveillance system. This will improve the country's ability to track and monitor resistance across sectors. The data generated from this project will strengthen the country's surveillance capacities, guide future monitoring programs, and assist the relevant directorates in making decisions regarding antimicrobial prescription and their waste disposal to ensure sustainable water reuse, and a comparative analysis between the environmental escape pathogens and information provided by institutions such as the NICD on clinical escape pathogens will assist in strengthening the knowledge regarding the clinical relevance of the strains detected in the environment, as well as dissemination of resistance organisms from one environment to another. I would like to acknowledge all of my supervisors and the bodies that are represented here, as well as the microbiology department. Thank you so much.
Right, thank you very much, Refile, for that presentation. Although we struggled to hear you, but thankfully most of the text was written in your slides. So thank you for that presentation. I am going to swiftly move on to the next presentation by Denzel Lakey. Are you here, Denzel?
I am.
And ready?
Madam Chair. If you don't mind, I won't use my video because my network is a bit unstable and it's a bit weak. So that's me, and I'll see if I can flag my presentation.
Right, I can see it.
Okay, right.
Please take it away.
Right, y'all. So I'm Denzel, and have this organization, ODCM, Organizational Development and Change Management. And I would like also to thank Professor Besaidenout for inviting me, because I think what I will be presenting on is actually very important. So let's just see if it goes into presentation mode.
It's already in presentation mode on my side.
Okay. Oh, right. It's just a bit on the slow side of life. It's not moving. Okay, right. There we go. Thank you. And also thank you to the new sound guys, because they're helping me with my technological challenges. Okay, next slide, please. Okay. That's the content of it. And for scientists, they like a little formula in between. So I'll throw that one in. And then I'll just go through some processes. And what it's actually all about is the observations from a different perspective, and that's a change management perspective, as to what's happened in the last few months, but also on a global platform, actually what we can do and how we should approach it, but what affected us. So we look at grief, we look at BCP and so on. And then I'll just give, towards the end, just an idea of what we could be looking at that will help us improve to get our return on investment. Next slide, please. Okay, so here are, that is Jack Canfield. He's the guy that wrote Chicken Soup for the, what's it, for the Soul, something like that. And if we know, if we just look at the circle on the right-hand side, it says there, arrow, which is events, reaction, and opportunity or outcome. But that happens every day in any context for individuals, for companies, and even on a global platform. So we know that. We know that you cannot stop these events from happening. It just happens. And if you look at the little frog inside there, that is the often used, that the frog don't even know that it's being cooked. So it just relaxes. And if we look at what's been happening to us, if we look at penicillin, for example, the resistance to penicillin is going on for a long time, and I've been listening to the themes coming out as the resistance, it's getting people to buy in. And when these things happen, how quick are we to respond to it, and do we address it seriously? If you listen to Dr. Barnes yesterday, she was saying, she spoke to the counselor and he just said, no vote, we're not voting for you. Sometimes it hits us like a bolt out of the blue, lightning, and it hits us hard. But however, despite the fact that we cannot control the event, we can control the reaction, how we react to that event that took place. And that reaction in itself is normally through our past experiences that we react, or the filters or our worldview, as some people would call it. However, but if you look at what guys like gurus and leadership like Maxwell would say, look at the end first. What would be like as an end result of this event that took place? And that we can discuss it, at length. And as the Japanese are saying, of course, for every event, the crisis, there's always an opportunity that we can actually take advantage of. Next slide, please. Okay, so maybe this is just a bit of, to contextualize what people has actually been going through. And Kubler-Ross actually had the 5 steps for the grieving process. And then Emerson Counseling came up with 7 steps, which I actually like, because if you look at this step number, people went into a state of shock. We know that, right? We didn't know if we were coming or going, and et cetera, et cetera. And the way we reacted, if you go back to the previous slide, the way we reacted was also maybe, could we have done better? Could we have done something differently? And okay, there we went back again, but okay, we're still working on the grieving process. The next slide, don't know why it reversed. Okay, there we go. And then also, just going through the simple, and I don't discuss this at length because we know we go through the denial process, people get very angry, even gender-based violence and domestic violence was blamed on COVID-19, and people were so close to one another that they actually just maybe realized who they really were. And then we go into a bargaining phase to see how, can we change things a little bit? Can we, benefit from it? Who's going to benefit from it? And depression is actually the crucial part there, because in any situation, when you go into depression, it's almost like a make or break situation that comes out of it. Make or break with a relationship, make or break with a project, and so on. And testing, and then of course just the acceptance part of it. That is when we say, okay, look, this is the situation. Now again, going to the outcome or the output, this is the situation. Let's see how we can benefit from it, how we can capitalize on it, because nobody goes into a situation and say, we want it to be the end, and go into it negatively. But we have to think about what do we want to achieve. Next slide, please. Okay, this is a topic on its own, but just to talk about it briefly, it's business continuity plan. How many people actually, how many organizations, how many individuals, how many families actually had a plan? People rush out to go buy gas. Organization says, oh, we need to have extra Wi-Fi, and or we need even universities for themselves in itself. Also, maybe we think of UNISA, maybe better position because they did distant learning. But other universities found out that most of these students actually work from the computer labs. They did Wi-Fi. I know some of the mobile companies came on board and gave Wi-Fi to students, to help them through. But was there a plan? And if there was a plan, how was that plan managed? Right? And we can think about that and say, we didn't have a plan. We just thought, okay, life will continue as it did yesterday. But we know the sun will rise, the sun will set. What are we going to do about it? The rain will come. And that is actually, and it says here in the circle part of it, it says that was a COVID-19 response. That's not a COVID-19 response only. That is actually a response that all organizations should have in place, and especially when you do your scenario planning and you say, if this fails, how else can we continue as usual, business as usual? Next slide, please. The next one is just the impact study. The business impact assessment. If we do not have this in place, or if we have this scenario happening, what will be the impact for us as an organization? Will we be able to continue with our business? Must we reassess our business plan? What do we need to have collaboration? I had lots of words of collaboration with others, etc. But the impact is, do we understand the functions of our business? Do we have a functional organigram of our business so that we know how to tweak it? Or do we just have the business as who's boss, who's second boss, and who are the laborers, the people that must produce the work? So that's another thing that we need to consider in our business, is the impact study of our business in the space that we operate in. Next slide, please. Okay, this is quite self-explanatory. To have just having discussed our business continuity plans, etc. Things happen. Sometimes we have to change in mid-flight. The show must go on. But just using the plane in itself, what was the first things that really happened? We all were grounded. We were all told to stay at home, and nobody could move anywhere. And yet we know that business had to go on. Services had to be delivered. And our hospitals were under stress and strain, and so were also the law enforcement people. But we know just that in retrospect, like they say, that's accurate science. Yeah, we could have maybe done things differently or better. Next slide, please. Okay, can you just click again, please? Now, if you look at this, this is a model that some companies use, and what it actually just says, the ADCAR, what it actually means at the bottom there, it's the awareness and so on, desire, knowledge, ability, reinforcement. And that is awareness of what has been happening. Did we understand actually what COVID-19 was all about? And how did we communicate that to the people affected? Now, we know it was a global, it was practically everybody was affected right through. But before you can actually bring that awareness, you must also actually think of who are your stakeholders? Are stakeholders there? Who is going to be impacted by what we're going to say? Because you cannot have a communication strategy in place if you don't know who you want to communicate with. And we'll talk about that maybe towards the end. But we know on the left-hand side, the axis there, a vertical axis, is that though any project starts off with a business plan, any project starts to have conceptualized, the design, and so on. Those are the steps that go through. And often projects only when they get to the implementation stage, then they find out, oopsie, we've missed out some people here. Next slide, please. Okay, that's your project plan, and that's its change management. And very seldom have I come across a project that has actually a change management strategy attached to it. We know the situation where we are, we know where we want to be, but what happens in between? And that is where the stakeholders come in. Who's going to make it happen? What must be changed? We know now on a global scale, you wear masks, wash your hands, sanitize, etc. We know that. But what change management does then is actually, how do we embrace this new style of living? What did we adopt it into our lives? And is it part of us? Did we use what is being given to us to be successful in what we are doing? The project design, that goes on. Design, develop, deliver. But what is being delivered and what is being delivered, are we using that? Because we heard of water today. We heard of AMRs. We heard of, how is it managed? Is it the role players and those affected by it? What are they doing about the situation and the information that we received? Next slide, please. Okay, so wrapping up now. How comprehensive did we do a stakeholder analysis in the project that we're doing? We spend sometimes millions, and I've been involved in some projects where it's multi-million rand projects, but they don't consider the stakeholders. There might be just one person there that has an influence on that particular project, where your road can't go straight through, the road bends. Why? Because that person wasn't consulted. Then when we do understand our stakeholders, we develop a communication strategy. What do we communicate? Who do we communicate with? And two, what do we want them to do with those stakeholders? Why are they stakeholders? What do we want them to do? And with that also, then we develop and design a change management approach. And it states an approach, which means that there are programs, there are strategies, there are projects within that. And we wouldn't like to discuss that in detail, but it is before the project starts, what happens during the project and the reinforcement of the project. Are we going to go back to our old ways of not wearing masks, not washing hands? And were we too complacent like the frog that, we use disinfectants and we use antiseptics just randomly, we use antibiotics just randomly. And then, of course, we heard so much about that yesterday also, that even antibiotics in mints, my goodness, that is a disaster. And then, of course, how do we measure what we are doing? What factors do we have to consider to say, look, we are moving forward, we are being successful, or we can improve in that area? So we have the scorecard, a criteria, a measurement of our projects and our programs. And then last but not least, the input and output, we know, right? You put ingredients in, you get a cake out, that's it. But what's the outcome of it? Is it being used? Right? And also, what's the impact then for the purpose that you've actually had that input in the first place? What was your business concept and what was your business imperative, so that you can also realize the return on investment? And that's actually the important part of it. Return on investment. We train so many scientists here, they don't get jobs because they're too highly qualified. What's our return on investment? And I do want to thank you again. Next slide. Thank you very much for inviting me. I know it's maybe a bit of a mouthful, but yeah, we can always chat about that again. And I will be available to maybe answer questions or to have further discussions offline. Thank you, Doc.
Right. Thank you very much, Denzel, for that presentation. Most certainly, yes, if time allowed, we would have chatted a little bit much further. Just to really maybe, so that maybe you can also help us contextualize our research, as you had some very valid questions there. So like I said, unfortunately, we have run out of time that was allocated for session 5. And I have to hand over to Dr. Lesego Molale-Tom to take us through session 6. But before I do that, I would really like to thank all the 6 speakers that have presented under session 5, and then also the researchers and students that have presented their work. Wish them all the best with what's remaining in their research. Right, we will meet again a little bit further on towards the end of the program, but for now, I would like then to hand over to Dr. Lesego Molale-Tom. Bye for now.
Thank you, Dr. Nonhlanhla, and thank you to all the presenters who presented thus far. We are now in session 6, and it will be running under the theme Antibiotics Stewardship, a Way Forward. Tom, and I will be introducing Mr. Henrico Hayes-Beck, who will be giving keynote address number 6. Henrico is a pharmacist who is passionate about therapeutic agents with a special interest in antimicrobials, over-the-counter solutions to everyday problems and medicine interactions. He also enjoys working with students and the challenges of teaching. He is a clinical preceptor who is responsible for the management of students in healthcare facilities in order to facilitate workplace learning. He's involved in the development and implementation of academic and practical content for workplace learning. He also serves as a local pharmacist at various pharmacies, and he gives special attention to medication interactions and antimicrobial stewardship, and a large portion of his time is currently spent advising people on the lifestyle changes necessary with the COVID-19 pandemic. Also, he is a health educator, and he loves giving lectures on various health-related issues to different audiences with a broad focus ranging from stroke awareness to viral infections. Welcome, Mr. Henrico.
Thanks, Sehu. I appreciate the invitation, and I'm sure that you guys have now listened to a lot of detailed information, but the question always remains what we can do as normal members of the public, not researchers, not academics, but what we can do. And for that, we need to take a step back and start from looking at everything in detail. All organisms, including us humans, are in a battle to survive. For us, luckily, we have developed the ability to change our environment to better suit our needs. For others, the rapid adaptation to their changing environment is needed to survive. Humans also found innovative ways to combat diseases, but the pathogens are starting to fight back, forming pockets of resistance, becoming ever more dangerous. So, before antibiotics, the world was a truly scary place. Diseases easily treatable today were almost always fatal. For example, 90% of all cases of bacterial meningitis were fatal, with survivors often showing lasting mental and functional disabilities. Strep throat often were fatal, and many middle ear infections showed brain involvement and mental decline. Treatments included bloodletting, leeches, and an assortment of concoctions, some of which had a basis in science, but many others simply need snake oil. This one actually steals the show, though. Is the toothache gone, or do you simply not worry about it anymore? So, a new weapon had to be found, and that's antibiotics. Alexander Fleming discovered penicillin by accident in 1928. By 1944, the mass production of benzyl penicillin was perfected, and widespread use of antibiotics followed. Previously incurable diseases are now curable. To quote Alexander Fleming, one sometimes finds what one is not looking for. When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medication by discovering the world's first antibiotic or bacteria, but I suppose that's exactly what I did. And he did revolutionize medicine. Unfortunately, with great power also comes great responsibility. In the years following the mass production and widespread use of penicillin, a boom in the discovery of antibacterial compounds followed. The time period between 1950 and 1960 was known as the golden age of antibiotics, and most of the therapeutic agents in use today were discovered in those 10 years. Unfortunately, that perceived power over microbial pathogens led to some questionable practices. Another worrying phenomenon is the lack of discovery of new antimicrobial classes over the past 30 years. So, what are we doing to endanger antibiotics and the efficacy of antibiotics? We're being responsible. During and shortly after the golden age of antibiotics, these bacterial killers were widely prescribed to nearly everyone and for all types of illnesses, regardless of the cause. It was widely used and still is today as a growth promoter in livestock, and due to its effectiveness, was promoted as a way to cure diseases previously avoided by lifestyle choices. Let's take a look at some of the old school adverts. For example, here, there are 3 antimicrobials suggested for poultry farming, namely erythromycin, tetracycline, nitrofenide, bactericidal agent, and sulfametazine, sulfonamide. Of these 3, the first and last fall into an antimicrobial class still used by doctors to treat illnesses. These practices expose environmental bacteria to these agents, causing resistance to these antimicrobials in organisms not normally found in humans, but paving the way for those resistant genes to be acquired by human pathogens. In the second advert, the widespread availability of penicillin is made known. This led to a false need for patients to ask for antimicrobials, and all following so-called wonder drugs, bacterial killers. Lifestyle, need I say more? So, the resistance. The Borg in Star Trek states that resistance is futile, but as the series proves time and time again, resistance is not futile. Normal cells, even pathologic bacteria, are always optimized to their environment. There are small differences in each cell. Without an antimicrobial present, susceptible cells might outcompete the resistant cells and thus outnumber cells that are slightly less efficient but with some resistance to antimicrobial agents. Once the antibiotic is introduced, most susceptible cells are killed off, leaving a higher concentration of resistant organisms, which, without the competition of the susceptible cells, will proliferate. In nature, without humans interfering and other artificial environmental pressures, the environment stays the same for long periods of time. However, as antimicrobial agents are introduced, the environment changes more rapidly and the organisms adapt to it on a faster rate, culminating in bacteria in the human body that has to compete with compounds designed to kill them. The scary part is, the horizontal transfer of genes may occur between organisms of different species, opening the door for a susceptible pathogen to gain resistance from non-harmful gut bacteria with established resistance due to long-term exposure to antimicrobial agents. Antibiotics. We are still in control, but with a very tenuous grips. Combination of antibiotics misuse and the amazing adaptability has given rise to a rapid spread of bacterial resistance. On your screens is a very, very scary story. MRSA, with methicillin resistance to Staphylococcus aureus, is now prevalent in more than 50% of all recorded in-hospital cases. Vancomycin-resistant Enterococci and fluoroquinolone-resistant Pseudomonas aeruginosa is becoming more prevalent. These organisms form part of the so-called escape group, which are naturally resistant to the first line of antibiotics. Luckily, there's still 1 or 2 antibiotics left to combat these pathogens, but the rise in resistance, the last line antimicrobials, and the lack of new antimicrobial agents that's effectively killing the microbe but safe enough to use in humans creates a very, very unstable grip on worldwide healthcare. So, how do our own use of medicine contribute to this crisis? Let's take a look. Some very unsettling prescribing practices, as we noted.
Nicole?
Yes.
We are unable to view your presentation.
Oh my shucks. Hold on. I'm going on here at a massive speed. Is this helping?
Yes, thank you.
Okay. I'm so sorry. You should have stopped me by the first slide, but let's go on. So antibiotics are prescribed for non-bacterial infections, and the justification given is prophylaxis for secondary infection, the high medicine safety of antibiotics, and so forth. Whilst combating secondary infection is necessary in some cases, this is definitely not true in all cases, least of all in upper respiratory tract viral infections. To limit the cost to the patient and time spent on simple cases, Let me just get my screen back up again here as well. Okay, never mind. To limit the cost to the patient and the time spent in simple infective cases, broad spectrum antibiotics are used in excess rather than the correct way of culturing a sample, identifying the bacterial pathogen and then performing susceptibility tests. This, however, isn't wrong in the eyes of the patient due to the impression that patients have that antibiotics cure all diseases. That last point goes hand in hand with the following. Most patients are under the impression that, as stated, antibiotics cure acute conditions, regardless of the pathogenic cause, be it viral, bacterial, or idiopathic. This false belief is a big driving force behind the overprescribing of antibiotics due to the fact that patients trust doctors who cure illnesses. Most diseases will clear themselves in roughly 10 to 14 days. This, combined with bad prescribing practice, forms a vicious circle of antibiotic overprescription. Added to this is a belief that non-recommended drugs are likely to help. COVID and the following shortage of chloroquine or the off-label use of ivermectin in South Africa is a prime example. The non-compliance of antimicrobial treatment length, regardless of the suitability of the treatment, is another concern. A study done in the UK stated that from roughly 1,000 patients with flu-like symptoms, 200 visit their doctor. This is fine, as visiting your healthcare provider will yield information on your illness or symptoms. More than 100 of those expect to be prescribed an antibiotic, and this is where the problem starts. 70 were prescribed antibiotics, but only 53 finished their course. What about the other 20 that didn't finish their course? Symptoms lessen when there's a reduction in the bacterial load of a patient, but too short a course of antibiotics will not fully eradicate all bacteria. It's conceivable the more resistant organisms will remain behind. We as a patient are the biggest reason that there's an overprescription of antibiotics in human health, as illustrated by the image on your screen. Doctors are constantly asked to prescribe antibiotics, and due to this, will likely prescribe it if they are unsure of the infection. Even doctors are sometimes forced to prescribe an antibiotic just to get rid of that pesky patient. In short, we need to be better patients, stop pressurizing doctors to prescribe us things that we think work, but might not be suitable. So, when we put everything together, what's the result? We are left with a perfect storm created by the rise in antibiotic resistance coupled with a reduction in antibiotic research. To best illustrate this, let's take a look at the major causes of death worldwide in 2014. Tetanus caused around about 60,000 deaths. Cholera, between 100 and 120,000 deaths. Diarrheal diseases, 1.4 million. Cancer, 8.2 million. Let's fast forward on antimicrobial resistance, which in 2014 caused about 700,000 deaths. If we continue the trend that we're following, there's going to be 10 million deaths. That's more than cancer and road accidents combined. The sad thing is, Africa and Asia will be hardest hit due to the majority of the population that has limited access to proper sanitation and running water. On a side note, we can see the same trend in antifungal and antiviral agents. Resistance developing during treatment is a much bigger problem in fungi than in viruses due to the extremely long treatment time required. Giving the fungal pathogen, yes, and it is less inclined to mutation than a virus, but there's still extra time to adapt to this new changing environment with an antifungal agent in it. Candida auris already shows some resistance against all 4 classes of antifungals, with a rising prevalence noted. Due to the faster life cycle of viruses and their propensity towards mutating, HIV is already resistant against all single drug treatment regimens, and a rise in multi-drug treatments are seen, and multi-drug treatment resistance are seen. Influenza is also showing increased resistance to oseltamivir, but luckily it's an extremely mutative virus. And this gives us a little sliver of hope with a noted rise in resistance just off the peak of oseltamivir prescription. This is in Europe, therefore the winter is opposite to our own. With the rise in oseltamivir prescription, it's also showing a rise in detection rate of resistance. But as the prescription rate of oseltamivir goes down over the summer, that resistance is also lost. And there's some evidence that some of the older antibiotics that has been out of general circulation for a few years now show better efficacy against the resistant organisms that caused their withdrawal in the first place. So, what can I as a pharmacist do? Well, I can educate public in dangers of antibiotic use, and that's what we're trying to do today. We can promote good hygiene practices, hand washing, surface disinfection, mask wearing. Luckily, we're now in a situation where that's the norm, not the exception of the rule. And I hope that that stays in place even after the eradication or the limitation of the COVID-19 pandemic. We can promote a wait and see prescribing trend regarding antibiotics. In other words, you get a prescription from a doctor with symptomatic as well as an antibiotic treatments prescribed. We'll try to advise the patient to take the symptomatic treatment first, give it a day or 3 to see if they feel better, and only then, if they're not feeling better, to come in and pick up the antibiotic. Once you have that antibiotic, don't stop the treatment early. Lastly, vaccinate. If you won't fall ill from certain illnesses, then antibiotics will be prescribed less. Remember, antibiotics don't work for everything. So if we continue using it like that, they might stop working when we need them most. In a more formal setting, pharmacists are also part of a multidisciplinary treatment team, assisting clinicians on selecting the correct antimicrobial agent based on susceptibility data of the organism. And this is where cultures and susceptibility tests against resistance now come in. As well as keeping in mind that the patient's already on certain medications and that some antibiotics may influence the metabolism or excretion of all the other medications as well. This is extremely important, as the most effective treatment over the shortest duration of time is the best way to limit the spread of antimicrobial resistance. So let's save the antibiotic. Let's not use it when it's not necessary. And when it is necessary, let's select the correct one. Antimicrobial stewardship is for everybody. It's not just for healthcare professionals. By taking the responsible route, each and every one of us as a member of public can help to spread the rise of antimicrobial resistance. So we need to educate ourselves on illnesses and how it's spread. Try to avoid antibiotics at first, and only to seek medical advice if it does not. We need to educate the others in this danger of antimicrobial resistance. And we need to keep in mind that everybody, not just doctors, pharmacists, nurses, are part of an antimicrobial stewardship team. Lastly, don't dispose of medication by flushing it. You're only giving organisms outside more things to feast on and become resistant against. Bring it to a pharmacy for safe destruction. There's just a few pointers. If you're ill, ask for help. Wash your hands, wash your working surfaces, and make sure that you continue the good hygiene practices that's now in place during the COVID-19 pandemic. So, what is antimicrobial stewardship? Something that's been thrown around now a few times. It's a coordinated program that promotes the appropriate use of antimicrobials, including antibiotics. It improves patient outcomes, reduces microbial resistance, and decreases the spread of infection caused by multi-drug resistant organisms. It's used in hospital to combat resistant organisms in theaters and in wards, but that definition must be expanded to include infection control and the spread of microbes around the home and in healthcare facilities. So keep calm and do antimicrobial stewardship, because everyone is part of an AMS team. Final thoughts. We didn't look for resistance, yet it was found. Multidisciplinary measures are necessary to combat antimicrobial resistance and the rise that we see in pathogens of that. We're running out of treatments to use. Future research in antimicrobial agents might employ the use of antibacterial viruses, also known as phages, metal-containing nanoparticles, to be used as a surface sterilizer in a liquid form, as well as a bacterial-resistant covering to use on surfaces. And then finally, to quote the father of all antibiotics, Alexander Fleming, it's not difficult to make microbes resistant to penicillin, and in the place of penicillin, we can put the word antibiotic in a laboratory by exposing them to concentrations not sufficient to kill them. The same thing has occasionally happened in the human body. And he said that in 1945 during his Nobel Prize lecture. And we see that the amount of truth that was encompassed in those words today. We are faced with a healthcare crisis, and we need to work together to get out of it. And there's just a couple of quick reads that you can take up if you're bored at night and want to fall asleep. Any questions?
Thank you, Enrico, for that thought-provoking presentation. And thank you for encouraging us to also be stewards of communicating and teaching the broader community about antibiotic resistance, not only in scientific terms, but also in a way that they can easily understand. And thank you for some of the advice that you gave us, such as the wait and see approach.
Thanks, appreciate.
I don't see any questions. Are there any questions for Enrico? Okay, it does not seem like we have any questions for now. Thank you very much.
Thank you.
So we move over to the closing plenary, and I will be introducing Prof. Ed Top. He is a native of Montreal, and he obtained his PhD from the Department of Microbiology at the University of Minnesota in 1988. Prof. Ed is a principal research scientist with Agriculture and Agri-Food Canada and has courtesy professorship appointments in the Department of Biology at the University of Western Ontario in London, Canada, and the Department of Soil and Water Sciences at the University of Florida in Gainesville. Prof. Ed's research concerns the interface between agriculture and human and environmental health, and he has generated over 260 co-authored publications. In the last decade, he has notably led several national studies concerning the fate and management of agroecosystems of pharmaceuticals and pathogenic as well as antibiotic-resistant bacteria carried in organic fertilizers of animal and human origin. He has organized a number of international workshops workshops and conference sessions concerning antibiotic resistance, agriculture, and the environment. He's the project coordinator for the Genomics Research and Development Initiative project on antimicrobial resistance, a key component of the innovation pillar in the Canadian federal framework for action on antimicrobial resistance. Prof. Ed is a member of the Science Advisory Board for JPI-AMR and a designated FAO/WHO expert for foodborne antimicrobial resistance. He's the past president of the Canadian Society of Microbiology microbiologists, recently received the AAFC Gold Harvest Award for Career Achievement, the Canadian Public Service Award for Excellence for Research Contributions, and was elected to the Academy of Agriculture in France. Prof. Ed, you are welcome.
Thank you so much for that introduction. Can you hear me?
Yes, we can, and we can see you as well.
Nice to be with you. Thanks so much for the invitation. Let me see if I can share my screen. Can you see the first slide here in the presentation?
Yes, we can, Prof.
Wow, technology works. So thanks so much for that introduction. I'm based out of Canada, but I happen to be coming to you today from Dijon and Burgundy in France through circumstances that unfolded that I hadn't anticipated. I'm actually on vacation this week visiting friends and colleagues here in France, where I've been working for many years now. So I was asked to say a few words or share a few thoughts on the environmental dimension of antimicrobial resistance, or environmental AMR, or what I may shorthand sometimes as EDAR, for environmental dimension of AMR. Let me see if I can get the next slide up. There we go. Can you see the next slide?
Yes, we can.
Okay, so for those of you who may not be familiar with or have never heard of London, Ontario, which is a long ways away from where you are now, this is a map of Canada at the top of the figure with the United States underneath. And if you look at the Great Lakes region, sort of in the center there towards the right, there's a triangular spit of land that goes from Toronto down to Detroit, and London is in that area there. So that'll just give you some idea of where I'm coming from and where my experience is. So I was asked to comment or share some ideas on two specific areas. One, what is happening in the global activity having to do with environmental AMR, and share that with you. And two, perhaps provide some ideas with what could be done in Africa in terms of research and potential interventions that would avoid the worst potential outcome from AMR. And I should say that much of what I'm going to say in the next few minutes is going to reiterate some of the thoughts that the previous speakers that I heard this afternoon said in their really excellent presentations. So pleased to have heard people this afternoon. So some context for those of you who may not be coming from the environmental AMR world. Why is environmental AMR something that we need to think about? Because it's within the context of One Health, which for the purposes of our conversation is really the idea that microorganisms have the potential to be exchanged between eukaryotes, whether they're people, animals, I could include plants in here. And that exchange is in every way, in every direction, and in many instances is mediated through the environment. And that is certainly especially the case with waterborne transmission of organisms that have antibiotic resistance, particularly in settings where water treatment and sanitation may not be as effective as one would like. So that's one of the two key ideas. It's that the environment is a vector or a medium through which AMR can readily be exchanged. The second idea is this hypothesis, which I think is quite well substantiated now, that the microbial world that's on the planet Earth, that was there long before we came along, something close to 4 billion years, many of those organisms carry genes that confer resistance to antibiotics that evolved in parallel with the ability to biosynthesize those antibiotics. And there's an enormous reservoir, coined the resistome, of genes in the environmental microbiome that potentially could be transferred and recruited and anchored into microorganisms that are of human or animal health concern. And that even though that happens infrequently, that ultimately is where genes that are ending up in the clinic are coming from. And that anthropogenic activities is potentiating that reservoir. So through, for example, input of chemicals that select or co-select for antibiotic resistance genes in environmental microorganisms, so the relative abundance of those genes may go up. Many activities, including environmental pollution with a variety of chemicals, can accelerate microbial evolution by through the effect on the SOS response, by increasing the frequency of conjugation, increasing the frequency of transduction, so that you have antibiotic resistance genes that are more likely to move around and therefore presumably more amenable to, at the end of the day, moving into human pathogens. So those would be the two general areas that those of us work in this area would be concerned about, and manipulating in some way to mitigate the relative role of the environment in the development of AMR. So what are some international developments that are relevant to this conversation? I would say the major one is, over the last few years, language in national action plans in most countries that I'm familiar with, and by global institutions. So for example, the United Nations Global Action Plan on AMR, language in the European Union, language that the ministers of health in the G20 countries have put out very recently, is all overtly One Health, both with respect to transmission of zoonoses and transmission of AMR. And you know, the zoonosis angle is complementary to the COVID-19 conversation as well. And so you see in this little cartoon here on the right, at the bottom right-hand side, this idea about One Health that has three pillars. The bottom right-hand corner, you've got agriculture. Bottom left-hand corner, you've got humans and human health. And at the top, you've got the environment more broadly. So that's, I think, very, very positive for us who work in the environment and need to get into fulsome conversations with the medical and veterinary community and have them understand that we need to consider the environment as well. That being said, I think it would be very fair to say that resources that are allocated to research, surveillance, mitigation steps, the funds in the human realm far outstrip those in the agricultural realm, that far outstrip those in the environmental realm. I hope I don't sound too bitter, but there's still some catching up to do in terms of actually resourcing the environmental dimension of AMR relative to the other ones. Another development are opportunities to fund international research and get different countries working with each other. And the Joint Programming Initiative on AMR, I think, is a very successful one and a very interesting model. So what it is essentially a pot where countries can fund their own researchers to work with people in other countries. So national funding institutions funding researchers who work through the JPI AMR framework together on themes that are related to AMR. And there's been a number of calls related to the environment transmission. I note that South Africa is a member of JPI AMR. As calls come up, different countries may agree or not to participate in those particular calls. I would note that South Africa has participated in the 2020 joint call on aquatic pollutants, which includes AMR, and I put the URL up there as well. So if you're not familiar with JPI AMR, I suggest you take a look at it. It might be an opportunity there for you. An area that is of significant interest, I think, in the international community right now is the relationship between environmental exposure to antibiotics or co-selective agents, biosolids, metals, potentially other kinds of chemicals, and the impact of that exposure on AMR in the environmental resistome. And the idea here is through a number of different lines of evidence that concentrations of antibiotics that are in some instances significantly below the minimum inhibitory concentrations and that are environmentally relevant are nevertheless such that they will confer a fitness advantage to organisms that carry genes that confer resistance to that antibiotic or class of antibiotics. So that means that organisms that are, let's say in the aquatic environment, that carry genes that confer resistance to an antibiotic that's, say, in a wastewater treatment plant effluent, have a fitness advantage. And so they will become relatively more abundant in that kind of environment. And so there's a lot of interest in exploring this, in establishing, in fact, what concentrations of any given antibiotic are safe, what aren't safe from this perspective. How could that information potentially inform standards or help mandate practice through evidence? I would say that internationally, that kind of information has not yet resulted in any water quality standards, for example, anywhere that I'm aware of. However, industry has proposed standards for antibiotic concentrations in effluents and receiving waters from pharmaceutical manufacturing operations. And this, of course, comes from evidence that was generated a number of years ago in the area around Hyderabad in India that has a high density of pharmaceutical manufacturing factories. And the concentrations of antibiotics that were leaving those factories in the effluents were enormous, exceeding in some cases minimum inhibitory concentrations. And so industry is sensitive to this, and based on evidence on predicted no effect concentrations and minimum selective concentrations through this paper here has suggested standards that would be appropriate and safe for wastewater treatment plants, for antibiotic manufacturing plants. And as I say here, that has not been implemented for wastewater treatment plants yet. Another area, and I would draw your attention to this recent report, is the relationship between environmental quality and food contamination as a route of transmission subsequently to people. And there was this working group with the European Food Safety Agency that came up with this report recently. I should say that it's very Eurocentric, very European-centric, but nevertheless, I think it still points out a number of risks of food pollution in primary production and processing that need to be evaluated in the particular circumstance that a particular country or region would find themselves in. Another area of international, I would say, excitement at the moment is wastewater surveillance for AMR, using either culture-dependent methods or probably more germane in the conversation now, culture-independent methods like metagenomic methods. And the idea here is the same one that people are using for the coronavirus, for COVID-19. It's going into raw sewage and making measurements in that sewage to determine what is being carried in the community that is shedding into that sewer shed. You can deploy the same kind of thinking to look at manure that's being generated from a herd, so that you don't need to surveil every, say, dairy cow on a large farm. You could look at the stored manure to get insights into what AMR that particular herd may be carrying. And then if you look at what is at the effluent from, say, the wastewater treatment plant, you can, following treatment, you can look at what is being emitted into the environment. And so this is, it's a relatively cheap way to get insights into what large numbers of people or animals are carrying. And there's interest in spinning this into some kind of global surveillance network. I'm part of a working group now with the United Nations Environmental Program, UNEP, that is developing a document sort of recommending what a UNEP might contribute to this environmental AMR problem. And there's a report currently intended to be released in February, and one of the recommendations will be a global surveillance network along the lines of what we're talking about here. Again, in the surveillance realm, a lot of interest in including environmental samples in integrated AMR surveillance systems, where say a country would implement surveillance to look at AMR in organisms that are carried by people, whether they're clinical organisms or otherwise, organisms that are in agriculture, whether it's food at retail, animals on the farm, processing plants, and the environment, to get a complete picture of AMR across that One Health continuum. The challenge for the environment and the discussion is what to measure and how to measure it, culture dependent, independent. Certainly in environments that are potentially subject to fecal contamination, whether it's an aquatic environment or a terrestrial environment, E. coli is going to be the gold standard, entirely appropriate. But what other culture or molecular targets might be of interest in environments, say, that are not subject to fecal contamination, but might be receiving, say, chemicals from antibiotic manufacturing plants. Another area, I think, of emerging interest is the use of antimicrobials as pesticides. And this is, I think, received relatively little interest. Here, the issue is in many instances, there's direct application of antimicrobials into plant-based food production systems to control plant diseases, whether they are bacterial or in particular fungal diseases. In some jurisdictions, antibiotics, so antibacterial agents, are used as well. This is not the case in Europe. It is the case in North America, and it's certainly the case in many other parts of the world. May or may not be well controlled. Here in Canada, we use streptomycin and kasugamycin for treatment of certain bacterial diseases of apples and pears, for example. In the United States, they do the same kind of thing. They're also using tetracyclines, I believe, to try to control greening disease, which is seriously damaging their citrus industry in the southern United States. On the fungicide front, this is really gaining international interest because of emerging filamentous fungal diseases such as Aspergillus fumigatus and yeast, so single-celled fungal diseases such as Candida aureus, which are becoming resistant to clinically relevant antifungal medicines, azole, other kinds of medicines. And the consequence of that is a very high rate of fatality in compromised patients who unfortunately acquire these infections. And the concern is that the agricultural use of fungicides, in particular the azole fungicides, where we're talking globally, millions of hectares of row crops, corn, maize, and wheat, for example, that receive megatons of these kinds of pesticides, and whether that is promoting mutations in fungi in the environment that at the end of the day are somehow ending up in the clinic. So this is an area of investigation and some regulatory concern at the moment. A major challenge in this realm is source attribution. And so this is a little bit more complicated cartoon concerning this One Health idea, just to point out the idea that, and this is a vast oversimplification, but you have multiple transmission routes between the various domains, the One Health domains. And here you see in the bottom right-hand corner, there's the human use of antibiotics. The bottom left-hand corner, there's terrestrial food animal production. At the top of the cartoon, there's that green oblong, so there's aquatic food production, and then the various other boxes largely represent terrestrial and aquatic environments, and receiving waste streams from people, waste streams from agriculture, direct entry of antibiotics. And the problem is, if a medical person asks me what percentage of human clinical AMR isolates come from the environment, what response do I give? If I'm asked by somebody like that, what percentage of human clinical isolates come from foodborne AMR? At the moment, it would be very difficult or impossible to give a solid answer. And so we have to fall back on the, I guess, common sense and the precautionary principle in most instances. So source attribution and gathering evidence to support what we're saying is an ongoing challenge. So the second question I was given here was what can be done in Africa in terms of research and potential intervention to avoid the worst pan or multi-drug resistance problem. Obviously, on this call, I'm arguably the least well-informed person in any position to offer people in Africa any advice on what to do. But I will say that I'm going to offer some generalities, which I think are applicable for anywhere. They're globally relevant. And I should say that, of course, Africa, even though I'm ignorant of the continent, is not one thing. It's a massive continent that has wide variation in culture, history, climate, landscape, finances, different ways of doing agriculture, et cetera, et cetera. And so solutions to mitigate AMR would have to be appropriate for particular regions or circumstances or sectors of the economy. So this just reiterates what I just said, which is I'm very humble in the context of this conversation. So some ideas here. So as I just said, where in Africa you've got pharmaceutical manufacturing, think about the emissions from those plants, look at them, think about standards. That would be an area of activity. Get better information on antibiotic concentrations in human and agricultural waste streams in the context of what I said about sub-MIC. So, what is being emitted into the environment in any part of Africa from human waste or from agricultural waste? And are those concentrations high enough that they would be of concern and that data would then inform change in practice, or would they not be of concern? Implement integrated surveillance systems as best as possible, given capacity, capability, resources. Implement wastewater AMR surveillance where possible in conjunction with the viral surveillance and in fact, other endpoints of human health concern that people excrete. Do what can be done to prevent AMR transmission from the environment to food, because foods like fresh fruits and vegetables, for example, represent an obvious direct route of human transmission to humans, and do that both in the primary production environment and in the processing environment. Use of antimicrobials, this idea of stewardship, which we've heard from the previous speaker, implement measures to keep animals and crops and people healthy such that antimicrobial use in animal and plant agriculture and the human realm can be reduced to the minimum required. So this is really the challenge. This is going to be where we need to go. It's reducing antimicrobial use right across that One Health continuum. In areas where water treatment and sanitation are absent or inadequate, investment into WASH would be a top priority, because particularly in lower income settings, anywhere in the world, if water treatment is inadequate, fecal contamination is going to be the primary route by which people get AMR, and that would be the top priority where you're going to get the biggest bang for your buck. Manage human and agricultural waste streams to protect the environment, and anticipate and prepare for climate change, doing a number of things, including creating more health challenges I guess with people, but certainly in agriculture, both plant and animal agriculture, that are potentially going to drive the use of more antimicrobials. Governance, improve oversight on AMU where you can, right across all stakeholders, and educate everybody. Again, just re-emphasizing what we heard in the previous talk. AMR is analogous to climate change. Global success is going to require local action. Don't think about fixing AMR across Africa. Think about local farming, local circumstances, what can be done to affect positive change, reducing AMU, reducing transmission that's appropriate given the realities of the local financial context, cultural, climate, anything else, circumstances that is relevant to whether or not what you want to achieve is going to be successful or not. So a couple of concluding comments. Further evidence to convince policymakers that environmental AMR is a problem relative to human and agricultural AMR and AMU is no doubt highly desirable, because we're always being asked, show me this is a real problem before I make an investment to try to fix it. However, given the currently available evidence, action on multiple fronts across that One Health continuum, including the environment one, is clearly called for. And with that, I would finish. Thank you very much for the opportunity to speak with you today.
Thank you, Prof. Ed, and thank you for sacrificing some of your holiday time to give us that very informative talk, and also some of the action plans and ideas that you've given us as a continent. Thank you very much. That was very insightful. Does anyone have questions for Prof. Ed? I don't see any questions in the chat box. Okay, if not, we will then move over to Dr. Nonhlanhla. Thank you, Prof. Ed.
Thank you again. Take care.
Thank you. Dr. Nonhlanhla will be then doing the closing remarks and closing off our two-day symposium.
All right, thank you very much, Dr. Molale Tom, and thank you to Prof. Ed Top for that. Really, I had like so many questions, but I just figured that I think probably maybe those, answering those questions might have to come with a little bit of some education for me. So maybe I would really ask that through the Northwest University, that at a later time we invite Prof. Ed Top in one of our events. As you know, that the WRC is also doing a lot of research on AMR. All right, so for this time that's been allocated to me, my job is to really give some remarks just for the sessions and this event that we've had for the past 2 days. So first of all, I want to thank all the participants that have been part of this 2-day event, both our speakers as well as the audience that has really made this such a very interactive engagement. And then I want to also thank the organizing committee for this event. So that includes the Northwest University, University of Johannesburg, Stellenbosch University, as well as the Water Research Commission. And then I just want to really thank the Northwest University as they were our hosts for this year's event. As you know that this event, it is an annual event as part of the World Antimicrobial Awareness Week celebrations. So I want to thank the Northwest University for their leadership, including the production team that has been really manning behind the scenes, making sure that all of us have our presentations up on time and that also we follow the program as it is. And then I also want to thank the Northwest University for also bringing along our esteemed guests. including Prof. Jeffrey Mpahlele, as well as our international guests. Today, we just had 2, and now we had just a third one in the form of Prof. Ed Top. Thank you very much for that. And then I just want to also once again extend sincere appreciation to the whole organizing committee for dedicating the time to be chairs for the sessions, and also those that presented some keynotes, imparted some really valuable knowledge, and also for encouraging their students or researchers within their units to also participate in this event. And then with that said, I also want to offer just some apologies. There were times where there were glitches. I mean, we have taken note of that, and certainly we will look into improving in the time, or is in the sound connection, or whatever it is, or it might be even the way that the themes were arranged. We look forward to hearing any feedback, if there is any, on how we can improve in future. Then I just want to just maybe briefly just put a spotlight on the Water Research Commission, our role as part of this event, and as well as a funding organization. So for those that are not familiar with who the WRC is, I would really maybe encourage you to maybe visit our website, www.wrc.org.za. So basically the WRC is a funding organization. It is a national research entity that is responsible for funding all water-related research in the country. So, I mean, I would then really encourage all those that maybe have some ideas from this today. If you are interested in putting in your research idea or proposal to the Water Research Commission, please do get in touch with us, and also look out for our annual call for proposals. So in terms of how the AMR research that is being funded by the WRC fits into our main research programs. So within the Water Research Commission, we have a water quality and health research program that is currently headed by Dr. Eunice Uboma-Jaswa, who is also here. So if you have any ideas, it would be good that you please discuss those ideas before you put them on paper. So, I mean, also as part of the presentations that we've listened to over this past 2 days, we have been taking notes furiously, and we have noted some of the gaps, research gaps, that most of you have highlighted in your research. So we would be very, very much really open to pursuing some of those gaps that you would have identified as a collaborator or as a funder to your research proposal. So like I said, that we have this program, and our annual call is in May, June every year. So, I mean, also, I mean, related to that, I have also had a little bit of talk on the need to really implement an environmental surveillance program on AMR. I'm so glad that also Refilwe earlier on spoke about it, and also recently, or just a few minutes ago, Prof. Ed Top also spoke about it as one of the recommendations that really need to happen. So, I mean, at the Water Research Commission, so what, I mean, maybe all of us, what we have learned maybe with the COVID is that it is really possible to implement a wastewater surveillance system where we can really analyze or check for a whole lot of other things, including AMR, COVID, or even some of the emerging contaminants, including pharmaceuticals and so on, and so other and many other things that really can be done under the wastewater-based epidemiology concept. So, I mean, at the moment, so we have a sort of a scoping study. So we are in the process of kickstarting a national AMR, or environmental AMR surveillance project or program. So at the moment, it is a scoping study that is led by the Northwest-University. But what the WRC will be doing in the next coming months is really now try to amass the capacity so that we can start building or putting in enough resources in the form of human and also laboratory resources that can contribute towards this environmental AMR surveillance program that we would want to put together. And then also, like I've already said that, so obviously what we normally do with WSC, if we have that need, we normally do some invite for proposals or through a call, terms of reference, so for people to join into the environmental surveillance network. So Prof. Ed Top also mentioned water JPI. So also from time to time, we collaborate with our international partners in co-funding certain areas that are also of priority in South Africa, mainly like water related, which are really run under the WRC. So for that also, in our webpage, there is a space where we normally post calls every now and then. So please do be on the lookout for such calls. Then finally, I just want to end with once again inviting everyone or institutions that were part of this year's event to please join in next year's event as part of the organizing committee or future hosts of a similar event. And by all indications, next year our host will be Stellenbosch University. So once again, then I would invite you to be on the lookout for next year's call, which will be led by Stellenbosch University. And yeah, look forward to interacting with you once again next year under the same topic or around the same time in November. But before then, I'm hoping that we can interact with some of you through our research meetings or through calls and so on and so forth. So that is that from my side. Once again, thank you very much, everyone. Thank you to our international guests, and thank you to our local participants, to our researchers, and also our audience for the questions and comments. And that is all from my side. I hope I have done justice. Thanks to you guys. You've been fantastic.
Thank you.
Bye.
Bye.
Bye, everybody.
Goodbye.
Goodbye.